Hello and welcome. I'm so grateful you're here. As we begin, I'd love it if you'd share in the comments where you're joining from and what time it is for you. It always amazes me to see how people connect from every corner of the world, morning or night. Thank you kindly to those who've subscribed and pressed that little like button. It really does help. And if you ever have requests or themes you'd like me to explore, please share them in the comments. Now, let's get started. Chapter 1. Understanding Synthetic Biology Fundamentals. Synthetic biology is an intricate and fascinating field that melds the principles of biology and engineering to design and construct new biological entities or systems. Imagine a world where we can create living organisms with specific functionalities, such as bacteria that produce sustainable fuels or plants that can thrive in arid climates. At its core, synthetic biology seeks not just to understand the building blocks of life, but to recompose and redefine them, pushing the boundaries of what is possible. To appreciate the scope of synthetic biology, it is essential to trace its roots back through the evolution of biotechnology. Biotechnology itself has a rich history, beginning with ancient practices like fermentation and selective breeding. These foundational techniques laid the groundwork for modern advancements. Fast forward to the mid-20th century, when significant breakthroughs in molecular biology unlocked the secrets of DNA. The discovery of the double helix by Watson and Crick marked a turning point, allowing scientists to delve deeper into the workings of genes and heredity. As technology progressed, so did our understanding of genetic engineering. The introduction of recombinant DNA technology in the 1970s enabled researchers to splice genes from one organism into another. This revolutionary technique paved the way for creating genetically modified organisms, or GMOs, which would go on to impact agriculture, medicine, and industry. The ability to manipulate genetic material has allowed for advancements in therapies for diseases and the production of insulin and other vital substances. In this context, the key concepts of DNA and genes become crucial. DNA, the molecule of life, carries the genetic instructions for the development, functioning, growth, and reproduction of all known living organisms. It is composed of nucleotides, which are the building blocks that form sequences, or genes. Each gene serves as a blueprint, coding for specific proteins that perform various functions within an organism. Imagine DNA as a library, where each book is a different gene, and the entire collection provides the instructions necessary for life itself. As the field of synthetic biology has matured, it has expanded its horizons beyond simple genetic modifications. Through sophisticated tools and methodologies, scientists are now capable of designing entirely new genetic circuits, akin to electrical circuits in engineering. These circuits can be programmed to perform complex tasks, such as detecting environmental pollutants or producing therapeutic agents in response to specific stimuli. The ability to engineer biological systems introduces a realm of possibilities that were once relegated to science fiction. Applications of synthetic biology are as diverse as they are groundbreaking. In medicine, synthetic biology holds promise for personalized therapies tailored to individual genetic profiles. Imagine treatments that are not just effective, but also uniquely suited to your genetic makeup, enhancing the efficacy of care. Beyond medicine, synthetic biology is poised to revolutionize agriculture by creating crops that can withstand extreme conditions, reducing reliance on chemical fertilizers and pesticides. This could lead to more sustainable farming practices and a reduction in the environmental impact of agriculture. Consider the potential of engineered microorganisms that can convert waste into valuable resources. These tiny organisms could be designed to break down pollutants or even generate energy, transforming how we approach waste management and energy production. The implications are profound, as they could help address some of the most pressing challenges of our time, such as climate change and resource scarcity. Moreover, synthetic biology touches on ecological restoration. By engineering plants that can thrive in degraded environments, scientists may be able to restore ecosystems and promote biodiversity. The vision of lush greenery reclaiming barren lands is an inspiring one. As we think about these applications, we might ask ourselves, what ethical considerations arise when we begin to manipulate life at such fundamental levels? The power to create life forms comes with responsibilities that cannot be overlooked. As we explore further, we should consider the voices of those who have shaped this field. One such figure is George Church, a pioneer in synthetic biology who once stated, "We can do things with biology that we never imagined." This sentiment encapsulates the essence of synthetic biology, a remarkable journey into the unknown, where the potential for innovation is limitless. The intersection of biology and technology continues to inspire and challenge us. With each advancement, we are pushed to reconsider our relationship with the natural world and the ethical frameworks that guide our scientific endeavors. The questions of what we can do and what we should do become increasingly intertwined. Synthetic biology opens a door to a future where we can design organisms that serve humanity's needs, address environmental issues, and enhance our quality of life. As we move deeper into this field, the exploration of these concepts will only grow richer and more intricate, revealing the depths of our understanding while beckoning us to ponder the responsibilities that accompany such power. Visualize a world where diseases are not just treated but prevented, where our food systems are resilient and sustainable, and where waste is transformed into resources. This vision is not just a dream, it is a horizon we are steadily moving toward, fueled by the innovations of synthetic biology. As we embark upon this journey through the fundamentals of synthetic biology, let us keep our minds open to the wonders and complexities that await. Imagine the possibilities as we continue to explore the intersections of life and technology, shaping the future of our planet and the lives of generations to come. At the heart of synthetic biology lies the remarkable capability to manipulate and construct genetic materials in ways that can revolutionize our understanding of life itself. This manipulation is not merely about altering existing organisms, but also about creating entirely new biological systems from scratch. Imagine a world where we can develop microorganisms that produce medicines or biofuels under specific conditions, organisms designed not just to survive, but to thrive in environments engineered for their success. The tools that enable this grand vision have evolved significantly over the years. Among the most transformative innovations is CRISPR-Cas9, a groundbreaking technology that allows for precise editing of DNA sequences. With CRISPR, scientists can target specific genes within an organism's genome, snipping and replacing them with remarkable accuracy. Jennifer Doudna and Emmanuel Charpentier, who pioneered this technology, emphasized the profound implications of their work when they said, "We can make changes to the genome of a living organism with unprecedented precision. This precision opens doors to correcting genetic disorders, enhancing crop resilience, and even creating organisms that can capture carbon dioxide from the atmosphere." As we consider the applications of these technologies, it is fascinating to delve into the concept of biological parts and systems. Just as engineers use standard components to build complex machines, synthetic biologists employ biological parts known as biobricks to assemble new functions within cells. Picture these biobricks as the Lego blocks of biology, each designed to perform a specific task. By combining these parts in innovative ways, scientists can construct living systems that respond dynamically to their environments, a concept that embodies the essence of synthetic biology. This flexibility and modularity also allow for a deeper understanding of biological networks. In nature, organisms interact within intricate networks of genes and proteins, forming feedback loops and control systems that maintain life. Synthetic biology seeks to replicate and even enhance these networks, enabling the design of organisms that can adapt and respond to changes in their surroundings. Envision a future where crops can sense and react to soil moisture levels, optimizing their growth in real time, leading to more efficient water usage and reduced irrigation needs. Synthetic biology's applications reach far beyond agriculture and medicine. In the realm of environmental science, engineered microorganisms can be crafted to digest plastics, or break down pollutants in oil spills. Imagine tiny bacteria designed to consume harmful substances, leaving behind clean water and soil. This approach not only highlights the potential of synthetic biology to address pressing environmental challenges, but also emphasizes the need for sustainable practices in an increasingly industrialized world. Moreover, the integration of synthetic biology with other scientific disciplines, such as materials, science, and information technology, is forging new pathways for innovation. The development of bio-inspired materials, which mimic the properties of biological systems, holds promise for creating more sustainable and efficient products. Think of textiles that can regulate temperature or materials that can heal themselves, inspired by the resilience found in nature. As we reflect on the capabilities of synthetic biology, we must also acknowledge the voices advocating for ethical considerations in this rapidly advancing field. The power to engineer life brings forth profound questions about ownership, consent, and ecological balance. As synthetic biologist Drew Endy articulated, we need to be careful about how we work with living systems. This cautionary sentiment urges us to be mindful of the implications of our actions, as the boundaries we tread upon are delicate and complex. Imagining the future of synthetic biology compels us to think about its role in fostering global sustainability. As the world grapples with climate change, food insecurity, and resource depletion, the innovations born from synthetic biology are positioned to offer solutions that could alleviate these challenges. Picture a world where sustainable practices are embedded in our very DNA, where the organisms we engineer not only coexist with nature, but enhance its resilience. In the medical field, the potential to create custom therapies tailored to individual genetic profiles raises exciting possibilities. The ability to produce living therapeutics, cells engineered to respond to and combat diseases, could redefine how we approach healthcare. This targeted approach aligns with the growing movement toward personalized medicine, where treatments are designed with the individual in mind, moving away from the one-size-fits-all paradigm. As we stand at this crossroads of possibility and responsibility, we are reminded of the importance of collaboration across disciplines. Scientists, ethicists, policymakers, and the public must engage in continuous dialogue to ensure that the advancements made in synthetic biology are guided by ethical frameworks. How do we balance innovation with caution? How do we ensure that the benefits of synthetic biology are shared equitably across society? The journey through synthetic biology is as much about understanding the science as it is about navigating the moral landscape that accompanies it. Each breakthrough invites us to ponder the implications of our creations, urging us to consider how our designs affect not only humanity, but the entire biosphere. As we continue to explore this captivating field, let us envision a future where synthetic biology serves as a bridge between human ingenuity and the natural world. With each advancement, we unlock new potentials, fostering a deeper appreciation for the complexity of life and the interconnectedness of all living systems. In this age of unprecedented technological advancement, the stories of those leading the charge, scientists, engineers, and visionaries, will shape the narrative of synthetic biology. Their work will inspire future generations to dream, to innovate, and to tread carefully as we redefine the boundaries of life itself. Let us carry this vision forward, embracing the wonders that lie ahead while remaining vigilant stewards of the world we inhabit. Chapter 2. Designing Life, Tools and Techniques. Imagine a world where we can orchestrate life itself, where the fundamental components of biology are not merely studied but actively designed and constructed. This vision becomes increasingly tangible through the remarkable tools and techniques that synthetic biology offers. As we explore the mechanisms of synthetic gene circuits, CRISPR technology, bioinformatics, and the construction of synthetic cells and organisms, we uncover a realm filled with both potential and responsibility. Synthetic gene circuits serve as the building blocks that enable the engineering of biological functions. These are intricate networks of genes that can be designed to perform specific tasks, much like electronic circuits in a computer. By combining various genetic elements, scientists can create circuits that respond to environmental signals in predetermined ways. For instance, researchers have developed circuits that allow bacteria to detect pollutants and respond by producing a visible signal, such as a change in color. This innovation can transform how we monitor environmental health, providing real-time insights into pollution levels. The beauty of synthetic gene circuits lies not only in their function but also in their modularity. Each component can be independently designed and tested, allowing for the assembly of complex systems from simpler parts. Imagine building a structure using Lego bricks, where each brick represents a different genetic function. This modularity facilitates rapid experimentation and innovation, leading to increasingly sophisticated designs and applications. However, the true marvel of modern synthetic biology becomes apparent with CRISPR technology. CRISPR, which stands for Clustered Regularly Interspaced Short Palindromic Repeats, has revolutionized the field of genome editing. This precise tool allows scientists to alter DNA sequences with unparalleled accuracy. By utilizing a guide RNA to target specific locations within the genome, scientists can create precise cuts in the DNA, enabling the addition or removal of genetic material. Consider the implications of this technology. The ability to edit genes opens doors to correcting genetic diseases, enhancing crop resilience, and even engineering organisms to produce valuable substances, such as pharmaceuticals or biofuels. Jennifer Doudna, one of the pioneers of CRISPR, aptly noted, "We can now rewrite the code of life." With this power comes the responsibility to tread carefully, balancing innovation with ethical considerations. As we embark on this journey through genomic landscapes, we begin to understand the importance of bioinformatics. This field merges biology with data analysis, enabling researchers to interpret vast amounts of biological data generated by modern sequencing technologies. Imagine navigating a vast library where every book contains a unique set of instructions for life. Bioinformatics serves as the librarian, organizing and analyzing these texts to reveal patterns and insights that drive our understanding of genetic information. Through bioinformatics, scientists can design new biological systems by predicting how genetic modifications will behave. This predictive capability enhances our ability to engineer organisms with specific traits, whether that be increased yield in crops or a microbe that can efficiently break down plastic waste. The integration of bioinformatics into synthetic biology exemplifies a powerful synergy, where data-driven insights lead to innovative biological designs. The culmination of these tools and techniques is evident in the construction of synthetic cells and organisms. The quest to create life from scratch has captivated scientists for decades. Synthetic cells, organisms engineered from non-living components, can be designed to mimic the functions of natural cells. These creations serve as valuable models for understanding basic biological processes and exploring new therapeutic avenues. One of the most ambitious endeavors in this realm was undertaken by the J. Craig Venter Institute, where scientists successfully created the first synthetic bacterial cell, named Mycoplasma mycoides, JCVI Syn 1.0. This landmark achievement demonstrated not only the feasibility of creating life from synthetic components, but also the potential to design organisms with tailored functionalities. As Venter himself stated, "We are in the process of creating a new form of life." As we venture further into this landscape of synthetic biology, we are reminded of the profound implications of our endeavors. The tools and techniques available to us grant unprecedented power over the very fabric of life. With each step forward, we must remain mindful of the ethical dimensions that accompany our innovations. In this ever-evolving field, we find ourselves on the precipice of a new era, where the boundaries of life are redefined through the lens of design and engineering. The journey through synthetic biology is not merely about creating new organisms, it is about understanding the intricate relationships that bind all living things. How do these elements interact within ecosystems? What unforeseen consequences might arise from our creations? As we reflect on these questions, we are drawn into a deeper understanding of the interconnectedness of life. Each synthetic organism we design must be harmonized with the existing biosphere, infusing our creations with a sense of responsibility toward the environment and society. Synthetic biology, in its essence, is a powerful reminder of the delicate balance between innovation and stewardship. As we continue to explore the tools and techniques available to us, we must embrace the wisdom of past scientists and thinkers who have paved the way for this remarkable field. Their insights serve as guiding stars, illuminating our path as we endeavor to design life itself. With the foundations established through synthetic gene circuits, CRISPR technology, bioinformatics, and the construction of synthetic cells, we are equipped to push the boundaries of what is possible. The canvas of life is vast and full of potential, inviting us to dream boldly and act wisely. What masterpieces await us just beyond the horizon? What systems can we design that not only enhance our lives but also nurture the earth itself? As we journey onward, let us carry with us the knowledge that we are not merely observers of life, but active participants in its design. We stand at a crossroads, ready to engage with the profound possibilities that synthetic biology presents, and it is our responsibility to tread thoughtfully into this uncharted territory. Synthetic gene circuits, CRISPR technology, and bioinformatics have illuminated our understanding of life, yet the pinnacle of this exploration is perhaps found in the construction of synthetic cells and organisms. This endeavor pushes the boundaries of our creativity and scientific prowess, challenging us to envision life crafted from the most fundamental elements. Imagine a laboratory where scientists meticulously design a cell as one might construct a piece of art. Each component, from membranes to metabolic pathways, is selected with intention, allowing for the creation of a biological system that can perform specific tasks. Synthetic cells can be designed to produce essential substances, such as insulin or vaccines, potentially transforming healthcare as we know it. These engineered systems serve as test beds for understanding cellular processes, offering insights into how life operates at its most fundamental level. One remarkable example of this ambitious undertaking is the creation of minimal cells. Researchers have sought to strip down cellular organisms to their essential components, revealing what is truly necessary for life to flourish. By studying these minimalistic structures, scientists are not only able to understand the core functions of life, but also explore how these functions can be repurposed for human benefit. Craig Venter's team has been instrumental in this pursuit, as they have explored the limits of life by constructing cells with only the most basic genetic instructions, akin to a blank canvas upon which new biological art can be painted. As we delve into the potential of synthetic cells, we encounter an important question: what defines life itself? Is it the ability to reproduce, to respond to stimuli, or to evolve? These inquiries spark philosophical debates as we consider the implications of creating life that may not conform to traditional definitions. The exploration of synthetic organisms compels us to rethink our understanding not only of biology, but of the very essence of existence. Creating synthetic life forms is not merely an academic exercise. It holds the promise of addressing some of the world's most pressing challenges. For instance, imagine a synthetic organism engineered specifically to consume carbon dioxide, effectively combating climate change. Research is underway to develop microorganisms that can convert greenhouse gases into benign or even useful products, such as biofuels. This intersection of synthetic biology and environmental stewardship exemplifies how innovation can harmonize with a commitment to protecting our planet. Moreover, the fusion of synthetic biology with other scientific disciplines has led to groundbreaking advancements. The integration of artificial intelligence into the design process enhances our ability to predict how synthetic organisms will behave in various environments. By simulating interactions and outcomes, researchers can refine their designs before they take the leap into the physical world. This convergence of disciplines exemplifies the collaborative spirit that drives innovation in synthetic biology, as scientists, engineers, and computer experts work together to expand the horizons of possibility. The ability to create life also raises critical ethical considerations. As we harness the power to design organisms, we must grapple with the consequences of our actions. The potential for unintended consequences looms large, prompting questions about the boundaries of intervention. How do we ensure that our creations coexist harmoniously with existing ecosystems? What safeguards must be put in place to prevent misuse of this powerful technology? These considerations echo the sentiments of renowned scientist Richard Feynman, who once said, "The imagination of nature is greater than the imagination of man." This reminder humbles us as we tread into uncharted territory, urging us to approach our endeavors with caution and respect for the natural world. As we continue to explore synthetic biology, we must embrace the concept of responsible innovation. Each synthetic organism we design should be carefully evaluated, not only for its intended purpose but also for its broader impact on the environment and society. This responsibility invites a dialogue among scientists, ethicists, and the public, fostering a collaborative approach to addressing the challenges and opportunities that lie ahead. In our pursuit of designing life, we are reminded of the intricate web that connects all living systems. The lessons learned from natural organisms inform our synthetic designs, leading to innovations that echo the elegance of evolution. Just as nature has honed its creations over millions of years, we too must learn from the past, building on the knowledge of those who have tread this path before us. As we look to the future of synthetic biology, we find ourselves not just as creators, but as custodians of the living world. The tools and techniques we wield grant us extraordinary power, yet with that power comes profound responsibility. The question we must continually ask ourselves is, how can we use this power to benefit all forms of life on Earth? Thus, as we push the boundaries of science and creativity, let us remain vigilant stewards of the ecosystems we inhabit. Let us design life not only with the ambition to innovate, but also with a commitment to nurturing the planet that sustains us. In this delicate balance lies the true artistry of synthetic biology, where the act of creation becomes a harmonious extension of the natural world, resonating with the rhythms of life itself. As we stand at the crossroads of science and ethics, the journey into synthetic biology unfolds with limitless possibilities. Each discovery, each engineered organism invites us deeper into the wonders of creation. The tapestry of life, both natural and synthetic, beckons us to explore, to learn, and to harmonize our innovations with the intricate dance of existence. What new realms of understanding await as we continue this journey? What life-affirming solutions lie just beyond the horizon? The answers are waiting to be discovered, and the adventure has only just begun. Chapter 3. Applications in Medicine, Healing with Biology. In the realm of medicine, synthetic biology emerges as a powerful ally, weaving together innovation and healing in ways once confined to the realm of science fiction. Imagine a world where diseases can be tackled not just with traditional pharmaceuticals, but with tailor-made solutions crafted through the intricacies of biological engineering. This chapter invites you to explore the profound applications of synthetic biology in medicine, highlighting the creation of synthetic vaccines, gene therapies, and biopharmaceuticals, all designed through the lens of biological ingenuity. The creation of synthetic vaccines stands as a testament to the possibilities of synthetic biology. In a world grappling with emerging infectious diseases, The ability to swiftly develop vaccines can mean the difference between containment and outbreak. Traditional vaccine development is often a time-consuming process, requiring extensive testing and production. However, synthetic biology offers a new approach, allowing scientists to design vaccines that target specific pathogens with precision. Consider the advent of mRNA vaccines. These vaccines use a snippet of genetic material to instruct cells in the body to produce a harmless piece of the virus, prompting an immune response. By employing synthetic biology techniques, researchers were able to develop these vaccines at an unprecedented speed during recent health crises, demonstrating not only efficiency but also adaptability. The success of mRNA vaccines in combating viruses has opened the door for their potential use against a range of diseases. Imagine vaccines tailored to target specific cancers or autoimmune disorders, harnessing the body's own defenses to fight back. Gene therapy, too, is a beacon of hope in the healing landscape of medicine. This approach seeks to correct genetic disorders by introducing, removing, or altering genetic material within a patient's cells. The concept might seem daunting, yet it holds incredible promise for individuals suffering from inherited conditions. For instance, the field has seen remarkable advancements in treating conditions like cystic fibrosis and hemophilia, where specific genes are targeted to restore normal function. One of the most notable successes in gene therapy comes from the case of a young boy diagnosed with a rare genetic disease known as bubble boy disease, or severe combined immunodeficiency. By using a modified virus to deliver a healthy copy of the gene responsible for the immune deficiency, doctors were able to restore his immune system, allowing him to live a normal life. This transformative approach not only highlights the intricate workings of our genetic code, but also underscores the profound impact that correcting a single gene can have on a person's life. The production of biopharmaceuticals through engineered organisms adds another layer to the healing capabilities of synthetic biology. Imagine microorganisms designed to produce life-saving drugs, such as insulin or monoclonal antibodies, at scale and with precision. This biomanufacturing process can significantly reduce costs and improve accessibility to essential medications. Take, for example, the use of yeast in producing human insulin. Scientists have engineered yeast cells to incorporate the human insulin gene, allowing these tiny organisms to synthesize this crucial hormone. The result is a more sustainable production method, reducing reliance on animal sources and enhancing the overall efficiency of drug manufacturing. This advancement not only improves the supply chain, but also opens the door for producing a wider array of complex therapeutics that were previously challenging to manufacture. Case studies of successful medical applications illustrate the transformative potential of synthetic biology. Beyond the stories of vaccines and gene therapies, we can look to the remarkable progress made in tissue engineering. Imagine being able to grow human tissues in the lab for use in regenerative medicine, a concept that was once a distant dream. Scientists have made strides in creating bioengineered tissues that can replace damaged organs or even be used in drug testing, reducing the need for animal models. Consider the heart tissue engineered from stem cells, which can be used to study heart diseases and test new treatments. This innovation not only accelerates the path to clinical applications, but also allows for a more ethical approach to medical research. As the field progresses, the possibility of growing entire organs for transplantation becomes increasingly tangible, addressing the pressing shortage of donor organs and offering hope to countless patients. The narrative of healing through synthetic biology is rich with stories of visionaries and pioneers who have dared to dream of a better future. One such figure is Jennifer Doudna, a pioneer in CRISPR technology, who has articulated the potential of gene editing by stating that we stand on the brink of a new era in medicine. Her work has inspired countless researchers to explore the implications of genetic engineering in treating diseases that were once deemed untreatable. As we reflect on these advancements, we are left to ponder the ethical dimensions of such powerful tools. With the ability to alter life at its very core, how do we ensure that these technologies are wielded responsibly? The balance of innovation and ethics becomes paramount in guiding the future of medicine. Imagine a future where diseases can be not only treated, but prevented through the thoughtful application of synthetic biology. What if we could foresee potential health issues in individuals before they manifest, enabling preemptive interventions? This vision is not far from reality, as the intersections of genomics and synthetic biology continue to unfold. As we explore the healing potential of synthetic biology in medicine, the stories of resilience and recovery intertwine with the science of life itself. Each advancement represents not just a leap in technology, but a profound impact on human lives, offering hope to those who seek healing in an ever-evolving landscape. The journey is just beginning, and the possibilities are as vast as the imagination of those who dare to dream. The landscape of medicine is rapidly evolving, and the applications of synthetic biology are poised to redefine our approach to health and healing. As we explore the remarkable potential of biopharmaceuticals, we encounter the fascinating world of engineered organisms, each designed to produce complex molecules that can lead to breakthrough treatments. Imagine a microscopic factory, a single-celled organism meticulously crafted to synthesize therapeutic proteins. This concept is more than just a dream. It is a reality in the realm of synthetic biology. Through meticulous genetic engineering, scientists can program bacteria, yeast, or even algae to produce valuable drugs. These organisms can be designed to express human proteins, such as antibodies, that play a crucial role in treating diseases like cancer and autoimmune disorders. The elegance of this process lies in its efficiency. By harnessing the natural capabilities of these organisms, researchers can create a scalable and cost-effective means of drug production. This innovation not only enhances the availability of crucial medications, but also reduces the environmental impact associated with traditional pharmaceutical manufacturing. One remarkable example is the development of monoclonal antibodies, which have revolutionized the treatment of various diseases. These antibodies are engineered to target specific antigens, making them powerful tools in combating conditions such as rheumatoid arthritis and certain types of cancer. The journey of monoclonal antibodies began in the 1970s, and today they represent a multi-billion-dollar industry, offering hope to millions. The production of biopharmaceuticals through engineered organisms also exemplifies the collaborative spirit of science. The contributions of remarkable individuals like Francis Arnold, a Nobel Prize-winning chemist, have illuminated the path towards using directed evolution to optimize enzymes in these production processes. Arnold's work underscores the beauty of nature's solutions, as she famously noted that nature is the best engineer. By mimicking evolutionary processes in the lab, scientists can create enzymes that work more efficiently, further enhancing the production of therapeutic compounds. As we delve deeper into the applications of synthetic biology, we arrive at the heart of gene therapy. This innovative approach not only seeks to treat the symptoms of genetic disorders, but aims to correct the underlying causes. The complexity of our genetic code is both a marvel and a challenge, and gene therapy represents a beacon of hope for those affected by inherited conditions. Consider the transformative journey of gene therapy in treating inherited blindness. A rare form of this condition, caused by mutations in a single gene, has been addressed through the introduction of a functional copy of that gene into the patient's retinal cells. This pioneering work has restored vision in patients who had previously been deemed irreversibly blind. Such success stories illuminate the profound impact that correcting a single genetic anomaly can have on an individual's quality of life. The challenges of gene therapy are not insignificant, however. Ethical considerations loom large as we ponder the implications of altering the genetic makeup of living beings. Who decides which genes are modified, and for what purposes? These questions are critical as we navigate the delicate balance between innovation and ethical responsibility in the realm of synthetic biology. As we reflect on the advancements made in this field, the stories of patients emerge, weaving a narrative of hope and resilience. Each case study serves as a powerful reminder of the human spirit's ability to overcome adversity. In a world where genetic disorders once dictated the course of lives, We now stand at the precipice of a new era where healing may lie within our grasp. The applications of synthetic biology in medicine extend beyond vaccines and gene therapies. The realm of tissue engineering presents another captivating frontier. Imagine not just repairing damaged tissues, but creating entirely new ones from scratch, tailored to the needs of individual patients. In recent years, scientists have made remarkable strides in creating bioengineered tissues, ranging from skin grafts used in burn victims to more complex structures like cartilage for joint repair. These innovations not only enhance the healing process, but also minimize the body's rejection response, as the tissues are often derived from the patient's own cells. The journey of tissue engineering is not without its challenges. The integration of engineered tissues into the body requires sophisticated techniques, including the development of vascular networks to supply nutrients and oxygen. Researchers are tirelessly exploring ways to create functional blood vessels within engineered tissues, a critical component for the success of future organ transplants. The vision of growing entire organs in the lab may still seem distant, yet the groundwork laid by pioneering scientists is promising. The possibility of addressing the shortage of donor organs could revolutionize transplant medicine, offering a lifeline to those waiting for a second chance at life. The stories of individuals who have benefited from these advancements remind us of the profound impact that synthetic biology can have. Imagine a child born with a congenital heart defect receiving a custom-engineered heart valve, or an elderly patient experiencing renewed mobility thanks to bioengineered cartilage. Each of these scenarios paints a picture of a future where synthetic biology not only addresses diseases, but restores the quality of life. As this chapter unfolds, we continue to witness the convergence of creativity, science, and compassion in the realm of medicine. The narrative of healing through synthetic biology is rich, filled with stories of those who dare to dream and those who benefit from their breakthroughs. Each advancement is a testament to human ingenuity and a reminder of the potential that lies ahead. In contemplating the future, we are invited to imagine a world where healthcare is not merely reactive but proactive, where diseases are anticipated and prevented before they take root. Through the lens of synthetic biology, we glimpse a horizon filled with possibilities, where the scientific and the human intertwine in a harmonious quest for healing. Chapter 4: Agriculture Revolution: Engineering Crops for the Future. The story of agriculture is one of humanity's oldest and most profound narratives, woven into the very fabric of civilization. As we stand at the crossroads of tradition and innovation, the agriculture revolution, driven by the principles of synthetic biology, invites us to reimagine the future of our crops. At the heart of this revolution lies the concept of genetically modified organisms, or GMOs, which have emerged as powerful tools in our quest to feed a growing global population. Imagine a world where crops are not merely products of nature, but are engineered with specific traits to enhance their productivity and resilience. This is the vision that GMOs bring to life. By altering the genetic makeup of plants, scientists can create varieties that are resistant to pests, diseases, and harsh environmental conditions. This technological advancement holds the promise of increasing yields while reducing the reliance on chemical pesticides and fertilizers. As the renowned biotechnologist Nina V. Fedorov once said, biotechnology is the most rapidly evolving field in science, and it is an essential part of the solution to global food security. The benefits of GMOs extend beyond mere productivity. They can also play a significant role in enhancing crop resilience to climate change. With the planet's climate becoming increasingly unpredictable, the ability to cultivate crops that withstand droughts, floods, and extreme temperatures is crucial. Consider the example of drought-tolerant corn, developed to thrive in areas suffering from water scarcity. These innovations not only safeguard crops but also help farmers adapt to changing climatic conditions, ensuring food security in the face of uncertainty. Yet, the story of agricultural innovation does not end with GMOs. The field of synthetic biology opens up new frontiers in pest control and sustainable farming practices. Imagine harnessing the power of nature itself to manage pests without relying on harmful chemicals. By employing synthetic biology techniques, scientists can engineer biological agents that specifically target pests, reducing the need for conventional pesticides. For instance, researchers have developed genetically modified bacteria that can produce substances toxic to certain insect pests while remaining harmless to beneficial species. This targeted approach not only protects crops, but also nurtures the delicate balance of our ecosystems. But with great innovation comes great responsibility. As we embrace these advancements, we must also consider the potential risks and ethical considerations surrounding GMOs and synthetic biology. What happens to biodiversity when we introduce genetically modified organisms into the environment? How do we ensure that these technologies are accessible and beneficial to all, rather than just a select few? These questions linger in the minds of scientists, policymakers, and the public alike. The potential risks associated with GMOs are often a topic of heated debate. Concerns about unintentional consequences, such as the possibility of gene flow to wild relatives or the development of pesticide-resistant pests, remind us that caution is essential. As agricultural scientist John W. D. Sutherland has noted, we must be careful that our innovations do not outpace our understanding of their ecological impacts. Ethical considerations also play a pivotal role in this discussion. The right to choose what we consume, the transparency of agricultural practices, and the implications for smallholder farmers are all crucial aspects that need to be addressed. The challenge lies in balancing the promise of technological advancements with the need for ethical stewardship. How do we ensure that the benefits of scientific innovation are equitably distributed? As we reflect on these themes, it is vital to remember that the agricultural revolution is not merely about technology. It is about humanity's relationship with the land. It is a reminder that our choices today will shape the agricultural landscapes of tomorrow. The stories of farmers and scientists working together to cultivate resilient crops are stories of hope and collaboration, where innovation meets tradition in a harmonious embrace. Imagine walking through a field of golden wheat, each stalk a testament to countless hours of research and dedication. Each grain represents not just sustenance, but the potential for a sustainable future. As we move forward, it is essential to foster a dialogue among scientists, farmers, and consumers, bridging the gap between innovation and understanding. Together, we can navigate the complexities of this new agricultural landscape, ensuring that it remains vibrant and diverse for generations to come. In this journey of exploration, we find ourselves at a pivotal moment in history. The tools of synthetic biology offer unprecedented opportunities to shape our agricultural practices, but they also challenge us to think critically about the implications of our choices. The agriculture revolution is not merely a chapter in the story of human progress. It is an ongoing narrative that invites us to imagine what is possible when we harness the power of science and creativity in service of the earth and its bounty. As we look ahead, let us embrace the potential of engineering crops for the future, remaining mindful of the ethical and ecological threads that intertwine our actions with the world around us. The path forward is illuminated by the promise of innovation, yet it is grounded in the wisdom of the past. With each seed sown, we have the opportunity to cultivate a future that nourishes not only our bodies, but also our planet and each other. As we immerse ourselves in the narrative of the agriculture revolution, we must acknowledge the interplay between technology and the environment. The cultivation of crops that are resilient to climate change is not merely a scientific endeavor. It is a critical response to the challenges our planet faces. Imagine fields of crops that sway gently in the breeze, their roots delving deep into the earth, equipped to withstand both drought and deluge. This vision is being realized through the remarkable advancements in plant genetics. One of the most captivating aspects of enhancing crop resilience lies in the exploration of plant pathways and their responses to environmental stressors. Scientists have identified specific genes responsible for drought tolerance in certain plant species. By applying genetic engineering techniques, researchers can transfer these beneficial traits to staple crops, ensuring they flourish even in the harshest conditions. This approach not only aims to increase agricultural productivity, but also to secure food sources for communities that face the stark realities of climate unpredictability. Consider the words of agricultural researcher Pamela Ronald, who emphasized, "We can use biotechnology to improve the resilience of crops to climate change." This sentiment captures the essence of harnessing scientific innovation to address pressing global issues. Through the lens of synthetic biology, we can visualize the potential of crops that not only survive but thrive under challenging circumstances. As we explore these innovations, we can also ponder the role of traditional breeding techniques alongside modern genetic engineering. Imagine a farmer standing in a lush field, utilizing both age-old knowledge and cutting-edge technology to cultivate a new variety of rice that can withstand saline water intrusion. This harmonious fusion of methods highlights the importance of integrating diverse approaches to achieve agricultural resilience. Turning our gaze towards sustainable farming practices, synthetic biology opens avenues for pest control that aligns with ecological principles. The traditional reliance on chemical pesticides has led to concerns about environmental degradation and the emergence of resistant pest populations. However, by engineering crops that can produce their own pest-repelling compounds, we can transform pest management into a more sustainable practice. Picture a tomato plant that emits natural repellents, keeping harmful insects at bay while encouraging beneficial pollinators to thrive. This method not only preserves the integrity of ecosystems, but also fosters a more sustainable approach to agriculture. Moreover, synthetic biology has the potential to revolutionize the production of biopesticides. By utilizing naturally occurring bacteria or fungi that target specific pests, researchers can develop products that minimize harm to non-target species and reduce chemical residues in our food systems. Imagine a world where farmers can confidently protect their crops, using solutions derived from nature itself, maintaining the delicate balance of life in their fields. Yet, as we navigate the intricate landscape of agricultural innovation, we must remain vigilant about the potential risks and ethical considerations that accompany these advancements. The introduction of genetically modified organisms poses questions about biodiversity and the long-term health of ecosystems. What happens when engineered crops interbreed with wild relatives? Will the introduction of synthetic biology disrupt the natural order, leading to unforeseen consequences? These concerns are echoed by many in the scientific community, reminding us that our innovations must be pursued with caution and respect for the complexity of life. The balance between progress and preservation is delicate. How can we ensure that our agricultural practices enhance biodiversity rather than diminish it? Ethical considerations extend beyond environmental impacts. They encompass issues of access, equity, and the rights of farmers. The potential for monopolization of agricultural technologies raises questions about who benefits from these advancements. Will smallholder farmers have the same access to genetically modified seeds as large agribusinesses? The challenge lies in creating frameworks that promote fairness and inclusivity in the adoption of new technologies. As we reflect on these themes, it becomes evident that the agriculture revolution is not solely about the science of crop engineering. It is also about nurturing a culture of responsibility and stewardship. The stories of farmers who embrace innovation while honoring their ancestral practices serve as powerful reminders of the importance of community and collaboration. Imagine local farmers gathering to share knowledge about sustainable practices, blending the wisdom of generations with the insights of cutting-edge research. In this context, the role of education becomes paramount. By fostering a deeper understanding of synthetic biology and its implications, we can empower consumers, farmers, and policymakers to make informed choices. Picture a classroom where students engage in hands-on experiments, exploring the principles of plant biology and the ethical dimensions of agricultural technology. Through education, we can cultivate a society that values both innovation and sustainability, ensuring that future generations inherit a world rich in agricultural diversity and resilience. As we traverse this landscape of possibilities, we must remain open to dialogue and collaboration. The path ahead is not without its challenges, but it is also filled with hope and potential. By embracing the insights of scientists, the wisdom of farmers, and the aspirations of communities, we can weave a narrative that honors both progress and tradition. With each seed sown, we are presented with an opportunity to reimagine our relationship with the earth. The agriculture revolution invites us to envision a future where technology and nature coexist harmoniously, a future that nourishes not only our bodies but also the delicate ecosystems that sustain us. Let us continue this journey, exploring the intersection of science and agriculture, as we work towards a future where food security and ecological health are not just ambitions, but realities. As we continue to explore the profound impact of synthetic biology on agriculture, we find ourselves drawn into the intricate relationship between crop engineering and the environment. The challenge of climate change has never been more pressing, and the need for resilient agricultural systems is paramount. Imagine a landscape where crops are not only productive, but are also guardians of biodiversity, helping to sustain ecosystems that are under threat. Enhancing crop resilience involves more than just genetic modification. It requires a holistic approach that considers the interplay between plants and their environments. One exciting area of research focuses on the development of crops that can utilize water more efficiently. Scientists are studying the mechanisms through which certain plants conserve water and adapt to arid conditions. By understanding these processes, it becomes possible to engineer crops that minimize water loss and maximize growth potential, ensuring that even in times of drought, the fields remain lush and fruitful. Consider the inspiring work of agricultural scientists who have dedicated their careers to addressing food insecurity. They strive to cultivate crops that can thrive in diverse and challenging environments. Their efforts remind us that innovation in agriculture is not solely about maximizing yield. It is also about nurturing the land and its resources. As one researcher noted, the future of agriculture lies in our ability to cultivate resilience in both crops and ecosystems. As we delve deeper into the role of synthetic biology in agriculture, we encounter the intriguing concept of engineered symbiosis. Imagine crops that can form beneficial relationships with soil microbes, enhancing nutrient uptake and resilience to pests. By modifying plants to interact more effectively with beneficial microorganisms, we can create a sustainable cycle of growth that minimizes the need for synthetic fertilizers. This approach not only bolsters crop health, but also enriches the soil, promoting long-term agricultural sustainability. Equally compelling is the notion of integrating traditional agricultural knowledge with modern scientific innovation. Picture a farmer utilizing time-honored practices alongside genetically engineered crops designed for enhanced resilience. This synthesis of old and new reflects a profound respect for the wisdom of those who have cultivated the land for generations. In this way, the agriculture revolution becomes not just a narrative of technological advancement, but also one of cultural preservation and appreciation. While we celebrate the advancements in crop engineering, we must also remain vigilant about the ethical dimensions of these innovations. The introduction of genetically modified organisms brings with it a responsibility to consider the ecological balance. How do we mitigate the risks associated with gene flow between modified and wild species? These questions compel us to adopt a precautionary approach, ensuring that we weigh the benefits of innovation against potential ecological consequences. The dialogue surrounding GMOs often stirs passionate opinions, reflecting the complexity of agricultural ethics. As we contemplate the future of food production, it becomes essential to engage with diverse perspectives, from scientists and farmers to consumers and policymakers. By fostering open discussions about the implications of synthetic biology in agriculture, we can cultivate a shared understanding that guides our collective decisions. Moreover, the potential for innovation in pest control cannot be overlooked. Imagine a future where crops are equipped with the natural ability to deter pests, reducing the need for chemical pesticides. This vision is becoming a reality through the application of synthetic biology. Researchers are developing crops that can produce specific proteins or compounds that repel harmful insects, maintaining the balance of ecosystems while protecting agricultural yields. However, as we embrace these advancements, we must remain aware of the potential risks associated with biopesticides. How do we ensure that these innovations do not inadvertently harm beneficial organisms? The challenge lies in refining our approaches and understanding the ecological ramifications of introducing modified crops into the environment. In this intricate dance of innovation and ethics, we find ourselves at a pivotal juncture. The agriculture revolution is not merely about engineering crops. It is a reflection of our relationship with the earth and our commitment to nurturing sustainable practices. By embracing the principles of stewardship, we can pave the way for a future where agriculture thrives in harmony with nature. Education plays a vital role in this journey. Imagine classrooms bustling with young minds eager to explore the wonders of plant biology and the ethical dimensions of agricultural technology. By empowering the next generation with knowledge, we can foster a society that embraces innovation while respecting the intricate web of life that sustains us. As we look ahead, the narrative of the agriculture revolution unfolds with promise. The stories of scientists, farmers, and communities intertwine, creating a rich mosaic of collaboration and creativity. Together, we can cultivate a future where the fruits of our labor nourish both people and the planet, ensuring that the legacy of agriculture endures for generations to come. In this evolving landscape, the potential of synthetic biology invites us to envision a world where technology and nature coexist, each enhancing the other. The journey continues, beckoning us to explore the depths of possibility and responsibility that lie ahead. With each passing day, we are reminded that the choices we make today will shape the agricultural landscapes of tomorrow, and it is our collective duty to ensure that they are vibrant, resilient, and sustainable. Chapter 5: Environmental Solutions, Bioremediation, and Sustainability. Imagine a landscape transformed by the power of synthetic biology, where the very organisms that populate our planet are enlisted to heal the wounds of our environment. The world is grappling with an increasing array of pollutants, from the remnants of industrial processes to the byproducts of human consumption. In this context, bioremediation emerges as a beacon of hope, where engineered microorganisms work tirelessly to cleanse contaminated soils and waters. These tiny yet mighty agents of change are capable of breaking down hazardous substances into harmless components, showcasing nature's ability to rectify its own imbalances when guided by human ingenuity. The process begins with the identification of specific strains of bacteria or fungi that possess the innate capability to metabolize toxic compounds. Scientists have harnessed these natural abilities, enhancing them through synthetic biology techniques, creating organisms that can thrive in environments previously inhospitable due to pollution. Imagine a bacterium that not only survives in oil-slicked waters, but actively consumes the hydrocarbons, converting them into benign substances. This is the essence of bioremediation, a collaboration between nature and technology, where the goal is to restore ecosystems to their former glory. Innovations in this field have already led to remarkable outcomes. For instance, the development of genetically modified bacteria capable of degrading heavy metals and other toxic wastes illustrates the potential of these living solutions. As researchers continue to explore the genetic blueprints of these microorganisms, they are building a repository of knowledge that empowers them to design even more effective agents for environmental restoration. Consider the words of a leading scientist in the field who remarked on the potential of synthetic biology. We have the tools to not only understand life, but to redesign it for our planet's needs. In addition to cleaning up existing pollutants, synthetic biology is also paving the way for the development of sustainable energy sources. The shift toward biofuels represents a significant stride in reducing reliance on fossil fuels, which are notorious for their environmental impact. Through the engineering of microorganisms, scientists have begun to produce biofuels from renewable sources, offering a glimpse into a future where energy is both sustainable and accessible. Imagine a world where algae, often viewed merely as a nuisance in water bodies, is cultivated for its ability to produce oil. This oil can be processed into biodiesel, providing an alternative to traditional diesel fuels. The process is not just a feat of engineering. It embodies the principles of sustainability by utilizing organisms that consume carbon dioxide and release oxygen during their growth. As they thrive, they are simultaneously addressing the very issue of carbon emissions that plague our atmosphere. This dual benefit encapsulates the harmony that synthetic biology aims to achieve with our environment. The potential for biofuels extends beyond algae. Engineered bacteria can also be designed to convert agricultural waste into fuel, transforming what would otherwise be discarded into valuable energy sources. This innovative approach not only mitigates waste, but also creates a circular economy within agricultural systems. It prompts us to ask, could our farming practices evolve to support both food production and energy generation? The answer lies in the creative application of synthetic biology, where every element of our ecosystem can be utilized efficiently. Carbon capture and sequestration represent another frontier in the fight against climate change, where synthetic biology plays a pivotal role. The objective is clear: to reduce the amount of carbon dioxide in the atmosphere, a greenhouse gas that contributes significantly to global warming. Imagine vast fields of engineered plants, each one a sentinel against climate change, capturing carbon through the process of photosynthesis and storing it safely in their roots or leaves. Scientists are exploring various methods to enhance the natural carbon-sequestering abilities of plants. By introducing specific genes that boost photosynthetic efficiency or increase root biomass, researchers are developing crops that not only contribute to food security, but also serve as effective carbon sinks. This dual function exemplifies the potential for synthetic biology to create solutions that address multiple global challenges simultaneously. The concept of carbon sequestration is not limited to plants alone. Microorganisms, such as certain bacteria and fungi, can also be harnessed for their ability to capture and store carbon. These organisms can be engineered to thrive in various environments, including those affected by climate change, further expanding the toolkit available for carbon management. In the words of a respected environmental scientist, the tools we need to combat climate change are already within our reach. We simply need to harness them wisely. This sentiment underscores the urgency of applying synthetic biology to environmental solutions. As we delve into the role of synthetic biology in biodiversity conservation, we encounter an intricate web of relationships where every organism plays a vital role in maintaining the balance of ecosystems. Biodiversity is not merely a collection of species. It is the foundation of resilience in our natural world. Yet, human activities have placed immense pressure on ecosystems, leading to habitat loss and species extinction. Synthetic biology holds promise in efforts to conserve biodiversity. Through the engineering of organisms, scientists can bolster populations of endangered species, enhancing their chances of survival in a rapidly changing world. Imagine a scenario where genetic tools are used to increase the reproductive success of a critically endangered plant species, allowing it to thrive once more in its native habitat. This is not a distant dream, but a tangible possibility grounded in the principles of synthetic biology. Moreover, synthetic biology can aid in the restoration of ecosystems. By reintroducing genetically diverse populations of species into their natural environments, we can enhance genetic resilience, making them more capable of withstanding environmental stresses. This approach emphasizes the importance of genetic variation in survival and adaptation, which is crucial for the longevity of any species. In addition to direct interventions, synthetic biology can also facilitate the monitoring of biodiversity. The development of biosensors, engineered organisms designed to detect changes in environmental conditions, can provide invaluable insights into ecosystem health. These living sensors can alert us to shifts in biodiversity, allowing for timely conservation efforts. Imagine walking through a forest where engineered plants signal the health of the ecosystem, providing an early warning of potential threats. As we reflect on the potential of synthetic biology to address pressing environmental issues, we find ourselves at a pivotal juncture. The synthesis of nature and technology opens doors to innovative solutions that can help heal our planet. The journey of bioremediation, sustainable energy, carbon capture, and biodiversity conservation illustrates the profound impact of human creativity when applied to the challenges we face. In this vast landscape of possibilities, the collaboration between scientists, policymakers, and communities becomes crucial. The path toward a sustainable future is not solely the responsibility of researchers. It requires the collective effort of all who share this planet. As we envision the future, we must ask ourselves how we can best utilize the tools of synthetic biology to create a harmonious existence, where technology works in tandem with nature, renewing and restoring the world around us. Imagine a future where the intricate dance of life is not only preserved but enhanced through the lens of synthetic biology. As we shift our focus to the role of synthetic biology in biodiversity conservation, we can begin to appreciate the delicate balance that sustains our ecosystems. Biodiversity acts as a life support system for the planet, underpinning food security, clean water, and healthy air. Yet, despite its vital importance, human activities continue to threaten this intricate web of life, leading to alarming rates of extinction and habitat degradation. In this context, synthetic biology offers innovative solutions that aim to safeguard and restore biodiversity. One of the most promising avenues is the genetic enhancement of endangered species. Imagine scientists deploying cutting-edge genetic tools to boost the reproductive success of a dwindling population of a unique butterfly species, ensuring that future generations can flourish in their natural habitats. This proactive approach reflects a shift from mere conservation to active participation in the evolutionary process, aiming to bolster the resilience of species facing unprecedented challenges. Consider the fascinating case of the California condor, once on the brink of extinction. Conservationists have utilized a combination of captive breeding programs and genetic management strategies to stabilize the population. By employing synthetic biology techniques, researchers can analyze the genetic diversity within this population, ensuring that the birds maintain a robust gene pool. This meticulous attention to genetic health not only aids in the survival of the condor, but also sets a precedent for similar efforts with other endangered species, illustrating the potential of synthetic biology to enhance conservation strategies. Furthermore, synthetic biology can facilitate the restoration of entire ecosystems. Imagine reintroducing a genetically diverse population of native plants into a degraded landscape. These plants, engineered for optimal growth and resilience, can restore soil health, promote water retention, and provide habitat for a variety of wildlife. Through this process, we not only revive the ecosystem, but also create a self-sustaining environment that can withstand future environmental pressures. This vision of restoration is grounded in the understanding that ecosystems thrive on diversity, and synthetic biology can be a powerful ally in nurturing that diversity. The application of synthetic biology extends beyond direct interventions in species and ecosystems. It also includes the development of advanced monitoring tools, such as biosensors capable of detecting changes in biodiversity. These engineered organisms can act as early warning systems, alerting us to shifts in species populations or the emergence of invasive species. Imagine walking through a vibrant forest, where these living sensors provide real-time data on ecosystem health, allowing conservationists to respond swiftly to emerging threats. This proactive approach transforms our ability to monitor and protect biodiversity, ensuring that we can act before a crisis unfolds. As the renowned ecologist once said, "The conservation of biodiversity is essential for the sustainability of life on Earth." This sentiment resonates deeply as we reflect on our collective responsibility to protect the delicate balance of our ecosystems. The integration of synthetic biology into conservation efforts not only enhances our understanding of biodiversity, but also empowers us to make informed decisions that impact the future of our planet. The intersection of synthetic biology and biodiversity conservation is a testament to human ingenuity and the potential for harmony with nature. It invites us to consider how we can harness our knowledge to cultivate a thriving planet. As we look to the future, we might ponder, how can we utilize these innovations not only to conserve, but to celebrate the rich tapestry of life that exists around us? The answer may lie in our willingness to embrace the possibilities that synthetic biology presents. In addition to its role in conservation, synthetic biology also fosters a deeper understanding of ecosystem interactions. By studying the genetic makeup of various organisms within an ecosystem, scientists can uncover the complex relationships that govern their coexistence. Imagine a thriving coral reef, where each species plays a crucial role in maintaining the delicate balance of life. Through synthetic biology, researchers can investigate the genetic underpinnings of these relationships, enhancing our ability to manage and protect these vital ecosystems. The benefits of such research extend to our understanding of ecosystem resilience. As scientists explore the genetic diversity within populations, they can identify traits that confer advantages in adapting to environmental changes. This knowledge allows for the development of strategies that enhance the resilience of ecosystems, ensuring that they can withstand disturbances such as climate change or habitat loss. Picture a world where ecological balance is not just a distant goal, but an achievable reality, bolstered by the principles of synthetic biology. Synthetic biology also promises to inspire a new generation of conservationists, fostering a sense of stewardship and responsibility toward our planet. By engaging young minds in the exploration of these technologies, we cultivate a culture of innovation and care for the environment. Imagine classrooms filled with students experimenting with genetic engineering to design organisms that can thrive in challenging conditions, fostering a sense of agency in their ability to affect change. This educational journey not only empowers individuals, but also ignites a collective passion for environmental stewardship, ensuring that future generations are equipped to address the challenges that lie ahead. As we reflect on the possibilities presented by synthetic biology in conservation and sustainability, we find ourselves at a remarkable crossroads. The integration of technology and nature opens up new avenues for collaboration, allowing us to work hand in hand with the natural world. It prompts us to consider the ethical implications of our actions and to strive for solutions that honor the intricate balance of life on Earth. Through this lens, we can envision a harmonious future where human creativity and the wisdom of nature coexist, nurturing a planet that thrives. In this unfolding narrative, the intersection of synthetic biology, bioremediation, and biodiversity conservation serves as a beacon of hope. As we explore these interconnected themes, we are reminded that the solutions we seek are not merely technological marvels, but are deeply rooted in our relationship with the world around us. The journey is one of understanding, respect, and a shared commitment to creating a sustainable future. Chapter 6: Industrial Biotechnology: Creating Sustainable Products. Imagine a world where the air is clean, the oceans are vibrant, and the products we use every day are not only effective, but also sustainable. This vision is becoming a reality through the fascinating realm of industrial biotechnology, where the principles of biology are employed to manufacture a diverse range of chemicals and materials. This biomanufacturing process breathes new life into our understanding of production, shifting from traditional methods that often rely heavily on fossil fuels to innovative approaches that harness the power of living organisms. At the heart of this transformation is the engineering of bacteria and other microorganisms. Picture tiny factories, invisible to the naked eye, working diligently to produce bio-based materials and chemicals. Scientists manipulate the genetic codes of these organisms, programming them to convert renewable resources into valuable products. This method is not merely a shift in technique, but a profound change in our approach to manufacturing. The notion of using biological systems to create everything from biodegradable plastics to biofuels challenges the very foundations of how we perceive production and sustainability. Consider the implications of such advancements. Traditional manufacturing processes often lead to significant carbon emissions and environmental degradation. In contrast, biomanufacturing utilizes renewable resources, reducing reliance on finite fossil fuels. This shift not only conserves natural resources, but also contributes to mitigating climate change. Imagine the satisfaction of knowing that the products you use every day are crafted through processes that are kinder to our planet. The potential of engineered bacteria extends far beyond just producing sustainable materials. These microorganisms can be tailored to synthesize complex compounds that are typically challenging to manufacture chemically. For instance, consider the production of bio-based plastics that can break down naturally after use. By harnessing the abilities of certain bacteria, we can create materials that serve their purpose without leaving a harmful mark on the environment. This innovation represents not just a technological breakthrough, but a pathway toward a circular economy, where waste is minimized and resources are reused. As we explore the landscape of sustainable alternatives to fossil fuels and plastics, we encounter remarkable figures who have shaped this field. One such pioneer is Frances Arnold, who was awarded the Nobel Prize for her work in directed evolution. Arnold's research focuses on designing enzymes that can facilitate the conversion of biomass into fuels and chemicals, effectively marrying biology with industrial applications. She emphasized the importance of innovation by stating, "We need to harness the power of evolution to solve humanity's greatest challenges." This sentiment embodies the spirit of industrial biotechnology, where evolving organisms can lead to groundbreaking solutions. Imagine a future where the fuels powering our vehicles and the materials comprising our products are not derived from the Earth's limited resources, but are instead cultivated from the very organisms that sustain us. This vision is becoming increasingly tangible. The production of biofuels, such as ethanol or biodiesel, showcases how engineered microorganisms can convert sugars and fats into energy sources, all while reducing greenhouse gas emissions. This is not just a theoretical concept. It is a thriving industry with the potential to reshape our energy landscape. As we delve deeper into the economic implications of industrial biotechnology, the market potential becomes increasingly evident. The demand for sustainable products is rising, driven by both consumer awareness and regulatory pressures. Companies that embrace biomanufacturing stand to gain a competitive edge in this evolving market. Imagine being part of a movement that not only prioritizes profit but also the health of our planet. The transition to bio-based products is not just an environmental imperative. It is also an economic opportunity that can lead to job creation and innovation. The market for bioplastics alone is projected to grow exponentially as consumers increasingly seek alternatives to conventional plastics. These bio-based materials, derived from renewable resources, not only reduce our carbon footprint, but also promote a sustainable lifestyle. Consider the excitement of being able to choose products that align with your values, knowing that each purchase supports a healthier planet. As we reflect on the innovations within industrial biotechnology, it is essential to recognize the intricate balance between technology and the natural world. While we engineer organisms to serve our needs, we must also ensure that we respect the ecosystems from which these solutions emerge. This mindfulness extends to the processes we develop and the materials we create, fostering a future where technology and nature coexist harmoniously. In this dynamic landscape, collaboration becomes a cornerstone for success. Scientists, engineers, and industry leaders must work together to drive the advancements that will shape our sustainable future. Imagine the power of collective innovation, where diverse perspectives converge to solve complex challenges. This collaborative spirit is crucial as we seek to harness the full potential of biomanufacturing, ensuring that we not only meet the needs of today, but also pave the way for future generations. As we contemplate the journey of industrial biotechnology, we find ourselves at a pivotal moment. The fusion of biology and engineering opens doors to possibilities previously thought unattainable. We are not just creating products, we are redefining the very essence of manufacturing. The stories of remarkable individuals, the groundbreaking research, and the emerging market for sustainable alternatives paint a picture of hope and possibility. Thus, the horizon is filled with promise, inviting us to explore further what other wonders await us as we continue to unravel the mysteries of living systems. As we embrace the principles of biomanufacturing, we embark on a path that not only honors nature, but also elevates our collective aspirations for a sustainable world. The implications of this journey extend far beyond the laboratory, promising a future where our choices reflect a deep respect for the planet and its resources. As we turn our attention to biomanufacturing, we find ourselves in a landscape rich with potential, where the transformation of raw materials into useful products is not only efficient but also thoughtfully designed to minimize environmental impact. This process utilizes microorganisms, such as bacteria and yeast, as biological factories. These tiny entities are adept at converting simple substrates, like sugars derived from plants, into complex chemicals, including those used in everyday products such as detergents, cosmetics, and biofuels. Imagine the simplicity of this process, a sugar molecule entering a bacterium, where a series of intricate biochemical reactions take place, ultimately yielding a substance that can be used for a variety of applications. This biological production is not merely a replication of natural processes, but rather a fine-tuning of them, where scientists can optimize pathways to increase yield and efficiency. A prominent figure in this realm is George Church, a visionary in the field of genetics and synthetic biology. He has long advocated for the power of engineered microbes, asserting, "We can use the tools of biotechnology to bring about a more sustainable future." This conviction is echoed in the advancements made over the years, as we learn to manipulate these microorganisms for greater precision and effectiveness. Consider the impact of these engineered bacteria on our energy needs. They can be designed to produce biofuels directly from agricultural waste, transforming what would otherwise be discarded into valuable energy sources. This not only alleviates waste but also provides a renewable alternative to fossil fuels, contributing to a more sustainable energy ecosystem. Imagine the far-reaching implications of a world where our energy is derived from waste, a closed-loop system that benefits both industry and environment alike. The exploration of sustainable alternatives extends into the field of bioplastics. Traditional plastics, derived from petroleum, contribute significantly to environmental pollution and resource depletion. In contrast, bioplastics made from renewable resources, such as cornstarch or sugarcane, offer a biodegradable solution that can reduce our dependency on fossil fuels. The process of creating these bioplastics is deeply intertwined with biomanufacturing, as the microorganisms involved can be engineered to enhance the properties of the materials produced. Visualize a future where packaging dissolves into harmless components, leaving no trace behind. This innovation presents not only an environmental benefit but also economic opportunities. The market for bioplastics is expanding rapidly, driven by consumer demand for sustainable products. Companies that pivot towards biomanufacturing are recognizing the dual advantage of contributing to ecological well-being while tapping into a burgeoning market. This transition is not just a moral choice, it is a strategic business decision. As we ponder the economic implications of these advancements, we can see a ripple effect throughout various industries. The biomanufacturing sector is poised to create new jobs, from research and development to production and distribution. This burgeoning field requires a skilled workforce, prompting educational institutions to adapt and offer programs tailored to these new technologies. The synergy between education, industry, and innovation becomes increasingly vital as we forge ahead in this new era. Furthermore, the potential for collaboration cannot be overstated. As different sectors come together, agriculture, energy, and manufacturing, a holistic approach emerges, fostering sustainable practices across the board. Imagine the power of a unified effort where scientists, entrepreneurs, and policymakers converge to create frameworks that support biomanufacturing initiatives. This collaboration can drive the policies needed to ensure sustainable practices are not only encouraged but incentivized. As we embrace this interconnectedness, we must also remain vigilant about the ethical considerations surrounding these innovations. The manipulation of living organisms raises questions about biodiversity, ecosystem integrity, and the long-term ramifications of releasing engineered organisms into the environment. It is crucial to approach these advancements with mindfulness, ensuring that our actions today do not compromise the health of our planet for future generations. The stories of success and innovation in industrial biotechnology serve as a reminder of what is possible when we harness the power of nature through scientific inquiry. Each breakthrough brings us one step closer to a world where our products are both effective and sustainable, where the choices we make reflect a commitment to preserving our environment. As we stand on the cusp of this exciting future, we are reminded of the profound responsibility that comes with such power. In the words of another pioneer in the field, synthetic biologist Drew Endy, we have to be careful not to try to rush to make the world better with synthetic biology that we forget to make sure it's safe. This guiding principle underscores the importance of not only innovation, but also ethical consideration in our pursuit of progress. In this journey of discovery, we are invited to imagine a world where the boundaries of biotechnology extend beyond mere production. We envision sustainable solutions that honor the delicate balance of nature while meeting the needs of our society. The potential for engineered organisms to create a positive impact on our environment and economy is vast, encouraging us to explore new possibilities and embrace a future filled with hope and promise. As we continue to delve into the realms of industrial biotechnology, we find ourselves not just as spectators, but as active participants in a transformative journey. Each advancement paves the way for a deeper understanding and appreciation of the role that biology can play in shaping our world. The narrative of industrial biotechnology is still being written, and we are all part of this unfolding story. Chapter 7. Ethical Considerations: Navigating Complex Questions. As we venture further into the intricate world of synthetic biology, we encounter a realm filled with not just the promise of innovation, but also the weight of ethical considerations. The potential to engineer life and manipulate biological systems invites us to confront profound dilemmas. What responsibilities do we bear when we create organisms that can alter ecosystems? How do we ensure that our advancements align with the values of society? In synthetic biology research, ethical dilemmas often arise at the intersection of science and morality. Imagine a scientist in a laboratory, working diligently to engineer a microorganism capable of breaking down pollutants in a contaminated river. The potential benefits are clear, yet questions linger. What if this engineered organism were to escape into the wild? Would it disrupt local ecosystems or outcompete native species? Such scenarios compel us to carefully consider the implications of our creations. Renowned biologist Paul Berg once remarked, "The most significant ethical questions are arising from the very science we are doing." This perspective serves as a reminder that with each groundbreaking discovery, we must rigorously examine the ethical frameworks guiding our work. The ability to design organisms with specific traits can lead to unforeseen consequences, raising questions about our right to intervene in natural processes. Public perception plays a crucial role in shaping the landscape of synthetic biology. As engineered organisms enter the marketplace, how do communities respond? Consider the reaction to genetically modified crops. While some embrace the potential for increased yields and reduced pesticide use, others express concerns about safety and the long-term effects on health and the environment. The divide in public opinion highlights the importance of transparent communication between scientists, policymakers, and the public. Imagine standing in a bustling farmer's market where one stall proudly displays organic produce and another showcases genetically modified crops. Each vendor shares their story, yet the narratives differ significantly. The organic farmer emphasizes tradition, sustainability, and a connection to the land, while the biotech representative highlights efficiency, innovation, and the potential to feed a growing population. This contrast invites us to ponder, how can we bridge the gap between differing viewpoints? How can we foster a dialogue that respects both scientific advancements and the values of those who may be cautious? Regulatory frameworks serve as the backbone for ensuring the safety and efficacy of synthetic biology applications. Agencies around the world grapple with the challenge of developing guidelines that balance innovation with precaution. Imagine a world where new technologies are subject to rigorous testing, ensuring they are safe for human health and the environment before they reach the market. This process, however, is not without its complexities. Different countries adopt varying approaches to regulation, leading to a patchwork of guidelines that can confuse researchers and the public alike. In some regions, stringent regulations may stifle innovation, while in others, lax oversight could jeopardize safety. The challenge lies in crafting policies that are both flexible enough to encourage scientific progress and robust enough to protect public interest. The ethical landscape extends beyond regulatory concerns. It encompasses the broader implications of our innovations. As we engineer organisms capable of performing tasks traditionally handled by nature, we must reflect on our role as stewards of the environment. The balance between innovation and ethical responsibility beckons us to consider the long-term impacts of our actions. What does it mean to innovate sustainably? Picture a landscape where engineered organisms not only contribute to human needs, but also enhance biodiversity rather than diminish it. This vision calls for a paradigm shift in how we approach synthetic biology. By prioritizing ecological integrity alongside technological advancement, we can work towards a future where progress and stewardship coexist harmoniously. In this delicate balancing act, the voices of ethicists, scientists, and the public are essential. Consider the insights of philosopher and bioethicist I. Glenn Cohen, who reminds us that the ethical implications of biotechnology are not merely theoretical. They are real, and we must engage with them. This engagement requires a commitment to ongoing dialogue, allowing diverse perspectives to shape the trajectory of synthetic biology. As we explore the societal impacts of engineered organisms, we uncover stories of hope and caution. Imagine communities that have embraced genetically modified crops, witnessing increased food security and economic stability. At the same time, we must acknowledge the voices of those who have experienced unintended consequences, such as loss of traditional farming practices or concerns about corporate control over seeds. Each narrative contributes to our understanding of the broader implications of synthetic biology. The urgency of addressing climate change further complicates the ethical landscape. Synthetic biology holds the potential to develop solutions such as carbon-capturing organisms or crops that require fewer resources. However, we must ask ourselves, are we simply using technology as a band-aid for deeper systemic issues? How do we ensure that our solutions are equitable and accessible to all communities? The concept of justice in innovation is crucial. By prioritizing the needs of marginalized communities and ensuring equitable access to the benefits of synthetic biology, we can foster a sense of shared responsibility. Imagine a future where the fruits of scientific progress are enjoyed by all, rather than a select few. This vision requires a commitment to inclusivity, transparency, and dialogue that acknowledges the diverse values of society. As we reflect on the regulatory frameworks guiding synthetic biology, we recognize the critical role they play in shaping public trust. Imagine a system where researchers are not only held accountable for safety, but also engage in open conversations with the communities affected by their work. This approach fosters trust and understanding, allowing scientific advancements to be integrated into society in a manner that respects ethical boundaries. The responsibility to safeguard the environment while advancing technology extends beyond borders. International collaboration becomes vital in addressing the global challenges posed by synthetic biology. Imagine scientists from different countries coming together, sharing knowledge, and working towards common goals. This spirit of cooperation can lead to innovative solutions that transcend national divides, enhancing our collective ability to tackle pressing issues such as food security and climate change. In navigating the complex questions surrounding synthetic biology, we find ourselves at a crossroads. The potential for innovation is immense, yet so too are the ethical considerations that accompany it. As we engineer life and reshape ecosystems, we must engage in thoughtful reflection and dialogue. By prioritizing ethics alongside innovation, we can chart a path forward that honors our responsibilities to both humanity and the planet. As we continue this exploration, let us remain mindful of the stories and voices that shape our understanding. Each discovery in synthetic biology invites us to reflect on our values, our choices, and the impact of our actions. How do we wish to be remembered in this era of transformation? What legacy do we want to leave for future generations? These questions linger, guiding us as we navigate this intricate landscape with care and intention. As we delve deeper into the ethical considerations surrounding synthetic biology, we must confront the ethical dilemmas that arise from the very act of creating new life forms. These dilemmas often stem from a fundamental question: who holds the power, and who bears the responsibility for the consequences of our innovations? This question resonates throughout the scientific community, prompting a reevaluation of our motivations and the potential repercussions of our work. Imagine a scenario where a groundbreaking organism has been engineered to produce an essential nutrient that could alleviate malnutrition in developing countries. The benefits are undeniable, yet the potential for unintended consequences remains. Could this organism unintentionally disrupt local agriculture or alter food webs in unforeseen ways? This highlights the necessity of ethical foresight in synthetic biology research. Each decision made within the lab can ripple outward, impacting ecosystems and communities far beyond the confines of our experiments. Ethical frameworks must evolve alongside scientific advancements, incorporating not only the perspectives of scientists, but also those of ethicists, sociologists, and the communities that may be affected. What if we included diverse voices at the initial stages of research? Engaging with stakeholders can cultivate a sense of shared ownership and responsibility, creating a collaborative environment where ethical considerations are woven into the very fabric of innovation. Public perception of engineered organisms also plays a vital role in shaping the ethical landscape. Imagine a community grappling with the introduction of a genetically modified organism designed to combat a local pest. While scientists tout its benefits, local farmers may harbor fears rooted in a lack of understanding or previous experiences with agricultural technologies. How do we bridge this divide? The answer lies in fostering transparent communication and education, allowing for an open dialogue where questions can be asked and concerns addressed. Consider the impact of social media in shaping public discourse. A single tweet can amplify fears or misconceptions about synthetic biology, leading to widespread misinformation. This reality underscores the importance of proactive engagement with the public, ensuring that accurate information is disseminated. Scientists must strive to be approachable, breaking down complex concepts into digestible narratives that resonate with everyday experiences. As we reflect on regulatory frameworks, we recognize that they must adapt to the rapid pace of innovation. Imagine a world where regulatory agencies are not seen as obstacles, but partners in the journey of scientific discovery. This partnership could facilitate the development of guidelines that are not only stringent but also flexible, allowing researchers to explore the full potential of synthetic biology while safeguarding public health and the environment. The ethical considerations extend to the very essence of what we seek to achieve through synthetic biology. Are we merely pursuing technological advancements for their own sake? Or are we genuinely committed to addressing the pressing challenges of our time? This question invites us to reflect on our intentions and the broader implications of our innovations. In the quest for sustainable solutions, we must remain vigilant against the temptation to prioritize short-term gains over long-term consequences. The concept of environmental justice emerges as a key consideration in this discourse. Imagine a future where synthetic biology is leveraged not only to develop solutions for climate change, but also to empower marginalized communities disproportionately affected by environmental degradation. By placing justice at the forefront of our innovations, we can create a more equitable landscape where the benefits of science are accessible to all. The ethical implications of synthetic biology are not confined to the present, they extend into the future as well. What kind of world do we want to create for generations to come? The decisions we make today will shape the trajectory of research and technology, influencing the relationship between humanity and the natural world. This long-term perspective compels us to engage in thoughtful reflection and to act with care. Consider the philosophical insights of those who have pondered the ethical dimensions of science. As we navigate the complexities of synthetic biology, we must ask ourselves, are we acting as responsible stewards of the planet, or are we merely exploiting its resources? It is through the lens of stewardship that we can align our scientific pursuits with the ethical principles that guide our actions. The emerging field of synthetic biology is a testament to human ingenuity and creativity, yet with great power comes great responsibility. As we engineer life and craft novel solutions, we must remain mindful of the ethical implications that accompany our actions. The journey ahead requires not only technical expertise, but also a commitment to ethical engagement, fostering a culture of responsibility that transcends the walls of the lab. As we consider the societal impacts of engineered organisms, we must remain attuned to the stories that unfold around us. Picture communities that have embraced synthetic biology innovations, witnessing transformative changes in their quality of life. Simultaneously, we must honor the narratives of those who have faced challenges, ensuring that their perspectives inform our understanding of the broader implications of our work. The urgency of climate change further complicates these ethical considerations. Synthetic biology offers solutions that could help mitigate its impacts, yet we must remain cautious. Are we relying solely on technology to address systemic issues that require holistic approaches? How can we ensure that our solutions are sustainable and equitable? These questions demand our attention as we navigate the intersection of innovation and ethics. As we reflect on the regulatory frameworks that govern synthetic biology, we recognize the need for ongoing dialogue among stakeholders. Imagine a world where scientists, policymakers, and communities collaborate to shape a regulatory environment that fosters innovation while prioritizing safety and ethical considerations. This vision invites us to consider the potential for a more integrated approach to governance, a model where diverse perspectives inform policy and practice. In this exploration of ethical considerations in synthetic biology, we find ourselves at a crucial juncture. The potential for innovation is immense, yet so too are the responsibilities that come with it. As we forge ahead, let us remain committed to engaging in meaningful dialogue, addressing the concerns of diverse stakeholders, and prioritizing ethical stewardship in our pursuit of scientific progress. Each step we take shapes the future, guiding us toward a more sustainable and just world. Chapter 8. The Future of Synthetic Biology, Trends and Innovations. Imagine stepping into a world where the boundaries of possibility expand before us, as emerging technologies reshape the landscape of synthetic biology. Here, the extraordinary potential for innovation is unfurling like petals on a blooming flower, revealing pathways that promise to transform industries, enhance sustainability, and redefine our relationship with nature. One of the most significant advancements lies in the realm of gene editing tools, which have reached unprecedented levels of precision and efficiency. With technologies like CRISPR-Cas9, scientists can now make targeted modifications to an organism's genome, allowing for the creation of biological systems with tailored functionalities. This precise editing capability opens doors to innovations that once seemed the stuff of dreams. Imagine a future where we can engineer microorganisms to produce essential pharmaceuticals more efficiently than ever before. These tiny powerhouses of biology could be harnessed to synthesize complex compounds, reducing reliance on traditional chemical synthesis methods that often yield significant waste. The implications for the pharmaceutical industry are profound. promising a new era in drug development that is both faster and more environmentally friendly. As we gaze into the future, it's crucial to consider how synthetic biology will extend beyond our planet. The exploration of space has long captured the human imagination, and synthetic biology is poised to play a pivotal role in this new frontier. The challenges of long-duration space missions require innovative solutions, particularly when it comes to sustaining human life in environments far removed from Earth. Imagine astronauts surviving on distant planets, relying on engineered organisms to produce food, oxygen, and even clean water. In this vision, synthetic biology becomes the backbone of extraterrestrial colonization. Scientists are already exploring how to use microbial life to create self-sustaining ecosystems in off-world habitats. For example, researchers are investigating the potential for algae to convert carbon dioxide into oxygen while providing nutritional sustenance. The beauty of these systems lies not only in their self-sufficiency, but also in their ability to adapt to harsh extraterrestrial conditions. The dream of colonizing Mars could become a reality, with synthetic biology acting as our guiding star. The convergence of artificial intelligence and synthetic biology is another frontier that beckons. Imagine the power of AI working alongside biologists, enhancing our understanding of complex biological systems and accelerating the pace of discovery. I can analyze vast datasets generated by synthetic biology experiments, identifying patterns and suggesting novel genetic modifications that may have otherwise gone unnoticed. This synergy has the potential to revolutionize the way we approach biological engineering, allowing us to harness the full power of data and computation in the design of living systems. As we look to the next decade, predictions for synthetic biology abound, offering glimpses of a world where technology and nature harmoniously coexist. The rise of bioinformatics will likely continue to reshape the field, enabling researchers to model and simulate biological systems with unprecedented accuracy. This will facilitate the design of organisms that can thrive in specific environments or produce desired compounds on demand. Imagine a future where bioengineered plants not only resist pests, but also sequester carbon, playing a role in climate change mitigation. Furthermore, synthetic biology will increasingly intersect with the fields of energy and materials science. The development of biofuels and biodegradable plastics will become more prevalent, as engineered organisms are designed to convert waste into valuable resources. This shift could transform our approach to waste management, leading to a circular economy where nothing is wasted and everything is repurposed. In this future, the ethical considerations surrounding synthetic biology will remain paramount. As we push the boundaries of what is possible, we must also reflect on the responsibilities that come with such power. The words of the influential biologist Richard Feynman resonate deeply. "What I cannot create, I do not understand." This sentiment urges us to approach synthetic biology with humility and respect for the intricate web of life we are seeking to manipulate. As we stand at the precipice of these exciting advancements, one must ponder the implications of our rapidly evolving capabilities. What does it mean for humanity to wield the power to design life? How do we ensure that our innovations serve the greater good, promoting sustainability and equity? The answers to these questions will shape the trajectory of synthetic biology and its impact on our world. In this exploration of the future, we envision a landscape rich with possibilities, where synthetic biology not only addresses the pressing challenges of our time, but also harmonizes with the natural world. Through creativity, collaboration, and ethical stewardship, the future of synthetic biology holds the promise of a more sustainable and vibrant planet, inviting us to dream boldly and act responsibly. The profound connection between synthetic biology and space exploration invites us to imagine a future where human life extends beyond Earth. The vast cosmic expanse, filled with challenges and opportunities, requires innovative solutions that synthetic biology is uniquely equipped to provide. Picture astronauts embarking on long missions to distant planets, carrying with them not just tools but living systems designed to thrive in alien environments. These systems could sustain human life by producing food, oxygen, and clean water through the manipulation of microorganisms and plants. As we contemplate the role of synthetic biology in space colonization, we can envision the creation of bioreactors capable of simulating Earth-like conditions. In these carefully engineered environments, genetically modified organisms could be deployed to recycle waste and generate vital resources. Imagine a scenario where astronauts cultivate crops inside a sealed habitat, using hydroponics and aquaponics systems, driven by microorganisms that convert waste into nutrients. This closed-loop system would not only support human life, but also serve as a model for sustainable living on Earth. The pioneering work of scientists like Robert Zubrin, who advocates for the use of in-situ resource utilization on Mars, underscores this potential. He imagines a future where colonists use local resources to create a self-sustaining ecosystem, reducing the need for costly supplies transported from Earth. By harnessing the power of synthetic biology, we can envision a thriving Martian colony where engineered organisms are the foundation of life, ushering in an era of interplanetary exploration that aligns with our aspirations for sustainability and resilience. The convergence of artificial intelligence and synthetic biology is another transformative trend that holds immense promise. As we delve deeper into the intricacies of biological systems, the role of AI becomes increasingly vital. Picture sophisticated algorithms analyzing vast amounts of genetic data, uncovering patterns that elude the human eye. This partnership between AI and biology enables researchers to simulate and predict the behavior of engineered organisms before they are even created. Imagine a scenario where an AI system proposes a novel genetic modification for a crop designed to withstand drought conditions. By analyzing historical climate data, soil profiles, and genetic information from diverse plant species, the AI could suggest targeted changes that maximize resilience. This synergy amplifies our capacity for innovation, allowing scientists to explore uncharted territories in genetic engineering with unprecedented speed and accuracy. One notable example of this convergence is the use of machine learning in drug discovery. AI algorithms sift through extensive databases of molecular structures, predicting which compounds are most likely to interact with specific biological targets. This accelerates the identification of potential therapeutics, transforming the landscape of pharmaceutical development. The implications are staggering. Imagine a future where we can develop new medications tailored to individual genetic profiles, enhancing efficacy and minimizing side effects. As we gaze into the horizon of synthetic biology, predictions for the next decade suggest a burgeoning field where technology will redefine our understanding of life itself. Bioinformatics will continue to evolve, enabling researchers to construct detailed models of biological systems. This computational prowess will allow scientists to design organisms that can respond to environmental changes, producing essential compounds on demand. Visualize a world where bioengineered organisms are cultivated in urban settings, producing food and biofuels while simultaneously capturing carbon dioxide from the atmosphere. These living factories could address food security and climate change, providing sustainable solutions that align with our global goals. The potential for urban agriculture, powered by synthetic biology, paints a vivid picture of cities teeming with life, where rooftops become gardens and walls become vertical farms. Moreover, the integration of synthetic biology into material science will likely lead to revolutionary breakthroughs. Imagine bioplastics created from engineered microorganisms that decompose naturally, reducing plastic waste in our environment. This shift towards bio-based materials not only addresses pressing ecological concerns, but also highlights the potential for a circular economy, where biological resources are continuously repurposed. As we venture into this promising future, the ethical dimensions of synthetic biology will remain a crucial aspect of our discourse. With the power to design life comes the responsibility to ensure our innovations align with societal values and priorities. The challenges of oversight and regulation may become more complex as the field advances, necessitating a robust framework that balances innovation with ethical considerations. Reflecting on the words of renowned geneticist Jennifer Doudna, who emphasizes the need for responsible stewardship in scientific exploration, we are reminded that as creators, we must be vigilant guardians of our creations. The questions we ask now about the implications of our discoveries, the risks we are willing to take, and the benefits we hope to achieve will shape the ethical landscape of synthetic biology as it propels us into a future rich with possibilities. As synthetic biology weaves its way into the fabric of our societies, we find ourselves at a pivotal juncture. The next decade promises to be a time of remarkable transformation, where the interplay of technology, biology, and human creativity will yield solutions to some of our most pressing challenges. In this unfolding narrative, we are not just spectators, but active participants, charting a course that blends innovation with responsibility and inviting future generations to dream of a world where life itself can be designed anew. The horizon of synthetic biology is further illuminated by the integration of advanced computational modeling and simulation techniques. Imagine being able to visualize complex biological interactions before they occur in the laboratory. This capability could not only expedite the design process, but also enhance our understanding of biological networks in their entirety. By employing sophisticated algorithms, researchers can simulate cellular responses to genetic modifications, providing insights that were previously unimaginable. This pathway toward predictive biology opens myriad avenues for innovation. For instance, consider the potential of creating synthetic organisms that can respond dynamically to environmental changes, such as variations in temperature or availability of nutrients. Rather than relying solely on trial and error in experimental settings, scientists could predict outcomes and refine their approaches in silico, saving both time and resources. This shift from empirical experimentation to computational foresight represents a significant leap forward in the field, potentially transforming how we develop solutions for agriculture, medicine, and environmental remediation. Moreover, as we explore these vast potentials, the connection between synthetic biology and social innovation becomes increasingly evident. Picture communities empowered by local biotechnologies, where individuals harness the principles of synthetic biology to address local challenges. From producing biofuels to creating sustainable agricultural practices, communities can cultivate resilience and independence. This democratization of biotechnology not only fosters innovation but also encourages a deeper engagement with the science that shapes their environment. In this landscape, educational initiatives play a crucial role. Imagine classrooms where students are not just passive recipients of knowledge, but active participants in the exploration of synthetic biology. Through hands-on experiences with microbial cultures and genetic engineering, the next generation can cultivate an understanding of biological systems that extends beyond the theoretical. As we contemplate the implications of these advancements, questions arise about accessibility and equity. Will the benefits of synthetic biology be universally shared, or will they remain confined to those with access to technology and education? The challenge lies in ensuring that the innovations of synthetic biology serve the greater good, addressing disparities while fostering a sustainable future for all. As we look toward the next decade, the synthesis of synthetic biology with other scientific disciplines will likely yield transformative results. Imagine breakthroughs in materials science, where engineered organisms produce textiles or construction materials that are not only functional but also biodegradable. These advancements could pave the way for a future where our consumption patterns no longer contribute to environmental degradation, but instead promote regeneration. The promise of synthetic biology in addressing climate change is particularly noteworthy. Envision microorganisms engineered to capture atmospheric carbon dioxide, converting it into useful products like biofuels or biodegradable plastics. This process not only mitigates the effects of climate change, but also transforms waste into valuable resources, embodying the principles of a circular economy. As we embrace these emerging trends, the narrative of synthetic biology unfolds as a story of collaboration. The confluence of interdisciplinary research brings together biologists, engineers, ethicists, and policymakers, all working toward a shared vision. Together, they navigate the complexities of innovation while remaining grounded in ethical considerations, ensuring that the advancements we pursue resonate with societal values. In this unfolding saga, we are reminded of the delicate balance between progress and responsibility. Each step forward in synthetic biology invites us to reflect on our role as stewards of the living world. The future we envision is not merely a collection of technological advancements. It is a commitment to fostering harmony between humanity and nature. The challenges of the coming decade will demand our creativity, resilience, and ethical foresight, ensuring that the innovations of synthetic biology enrich our lives while protecting the delicate ecosystems that sustain us all. Chapter 9. Global Collaboration: Advancing Synthetic Biology Together. In the evolving landscape of synthetic biology, the importance of international cooperation in research cannot be overstated. Imagine a world where scientists from diverse backgrounds and cultures come together, sharing knowledge and resources to tackle some of humanity's most pressing challenges. This collaborative spirit fosters innovation and accelerates discoveries, creating a rich environment where ideas flourish. Consider the Global Plant Council, an initiative that unites experts from around the world to advance plant science and biotechnology. By pooling their insights and expertise, these researchers work collaboratively to enhance global food security and address climate change. This partnership exemplifies how collective efforts can yield remarkable advancements in understanding plant biology and improving agricultural practices. Another stunning example of international collaboration is the Synthetic Biology 2.0 movement, which aims to democratize access to synthetic biology tools and resources. It emphasizes the importance of open-source biology, where researchers share their findings, methodologies, and technologies with the broader community. This shift towards openness encourages community-driven innovation, allowing individuals and small teams to contribute to groundbreaking discoveries. Imagine scientists in different corners of the globe exchanging ideas and protocols, leading to a surge of creativity and progress in synthetic biology. Open-source biology is not merely a concept. It represents a profound cultural shift in how scientific knowledge is shared and developed. By removing barriers to access, researchers can build upon each other's work, accelerating the pace of innovation. As the renowned scientist and advocate for open science Jennifer Doudna passionately stated, "When we work together, we can achieve more than we ever thought possible." The impact of open-source initiatives can be seen in various global synthetic biology projects. One notable example is the IGEM competition, which invites teams from around the world to design and build synthetic biology projects. These projects often address local and global challenges, from developing biosensors to detect environmental pollutants, to engineering bacteria that can produce biofuels. The collaborative nature of IGEM fosters an environment where young scientists can experiment, learn, and inspire one another, creating a vibrant community dedicated to advancing the field. As we explore further, it becomes evident that while international cooperation brings immense potential, it also presents challenges. The governance of synthetic biology at a global scale requires careful consideration of ethical, legal, and environmental implications. How do we ensure that the advancements in synthetic biology are safe, equitable, and sustainable? These questions resonate deeply, as they compel us to examine the frameworks that guide our scientific endeavors. One of the significant challenges lies in establishing regulatory standards that can be universally accepted. Different countries may have varying approaches to biotechnology, leading to discrepancies in safety assessments and ethical considerations. This lack of consensus can hinder the progress of international collaborations. Imagine a scenario where a groundbreaking synthetic organism developed in one country faces restrictions in another due to differing regulations. Such challenges urge us to find common ground and develop global policies that prioritize safety while fostering innovation. Moreover, the rapid pace of advancements in synthetic biology often outstrips existing regulatory frameworks. What happens when the technology evolves faster than our ability to govern it? This reality calls for proactive discussions among stakeholders, including scientists, policymakers, and ethicists, to ensure that the benefits of synthetic biology are harnessed responsibly. The opportunities within this landscape are equally compelling. By engaging in global governance discussions, we can shape a future where synthetic biology is used ethically and effectively across borders. Collaborative platforms, such as the World Health Organization's efforts to facilitate the sharing of genetic data for vaccine development, showcase how coordinated action can lead to significant advancements in public health. In addition, the rise of global networks dedicated to synthetic biology research fosters an environment where shared knowledge can flourish. Initiatives like the International Synthetic Biology Research Center aim to bring together researchers and institutions from various countries, promoting the exchange of ideas and best practices. This collective effort not only strengthens the field, but also cultivates a sense of unity and purpose among scientists dedicated to addressing global challenges. As we contemplate the future of synthetic biology, it is crucial to recognize the potential for collaboration to drive meaningful change. The interconnectedness of our world means that the challenges we face are often shared, transcending geographical and cultural boundaries. By fostering international cooperation, we can harness the collective intelligence of the global community, propelling advancements that benefit all of humanity. Imagine a future where synthetic biology projects are not confined to national borders, but are instead embraced globally, where discoveries made in one corner of the world can improve lives in another. This vision is not merely a dream. It is a possibility waiting to be realized through concerted efforts and shared aspirations. As we continue to explore the extraordinary realm of synthetic biology, we must remain vigilant in our commitment to ethical practices and global collaboration. The potential for transformation lies in our hands, guided by the understanding that together we can navigate the complexities of this field and unlock its boundless possibilities. In this journey, we celebrate the voices of those who have championed collaboration in science. As the esteemed biologist E.O. Wilson once expressed, "We are not only the product of our ancestors' actions, but also the architects of our own future." This sentiment echoes through the halls of synthetic biology, reminding us that our collective efforts can sculpt a brighter, more sustainable world. Through the lens of global collaboration, we can illuminate the path toward innovations that resonate beyond borders, addressing the intricate web of challenges that confront our planet. The future of synthetic biology is a shared narrative, one that beckons us to partake in its unfolding story, contributing our unique perspectives while learning from one another. As we embrace this global tapestry of knowledge and creativity, the potential for breakthroughs becomes limitless. What lies ahead in the realm of synthetic biology is not just a testament to individual brilliance, but a celebration of our shared commitment to advancing science for the betterment of all. The journey of global collaboration in synthetic biology is not without its hurdles, yet these challenges present opportunities for growth and innovation. As nations come together, the complexity of differing regulatory landscapes can initially seem daunting. However, it is essential to view these variations not just as obstacles, but as a chance to forge a more robust framework for governance. Each country brings its own set of experiences, ethical standards, and scientific priorities to the table, creating a rich dialogue that can lead to more comprehensive and inclusive regulations. Imagine a roundtable of scientists, policymakers, and ethicists from diverse cultures, each sharing their perspectives on the safe deployment of synthetic biology. The conversation flows, filled with the vibrancy of different viewpoints, aiming to establish guidelines that foster innovation while ensuring public safety. This collaborative effort can lead to the development of international standards that not only address safety and efficacy, but also respect the unique values and needs of different societies. The establishment of global partnerships can also enhance the capacity for scientific research. By pooling resources, countries can tackle larger projects that may be beyond the reach of individual nations. For instance, consider large-scale initiatives like the Earth Biogenome Project, which aims to sequence the genomes of all known eukaryotic species. This ambitious collaboration requires the combined efforts of scientists across continents, each contributing their expertise and technology. The result is a monumental leap forward in our understanding of biodiversity, offering insights that can guide conservation efforts and sustainable practices worldwide. Additionally, the role of technology in facilitating global collaboration cannot be overlooked. Digital platforms enable researchers to connect, share data, and collaborate in real time, regardless of geographical barriers. Online repositories for scientific data, such as the Open Biological and Molecular Data Network, exemplify how technology can democratize access to information, allowing scientists from developing countries to participate fully in global discussions. As we consider the implications of these global networks, we must also acknowledge the potential for community-driven innovation. Local communities, empowered by access to synthetic biology tools, can address specific challenges in their regions. Imagine a group of researchers in a small island nation, utilizing open-source biotechnologies to develop salt-resistant crops that thrive in their unique environment. These grassroots projects highlight the power of local knowledge combined with global resources, creating tailored solutions that acknowledge and respect regional biodiversity. Furthermore, the success of such initiatives underscores the importance of education and capacity building in synthetic biology. By investing in training programs and educational resources, countries can cultivate a new generation of scientists equipped to contribute to global collaborations. Initiatives like the BioBuilder Educational Foundation provide students with the opportunity to engage with synthetic biology concepts, sparking interest and fostering innovation from a young age. This emphasis on education not only prepares future scientists, but also builds a community that values collaboration and shared knowledge. While the prospect of global collaboration in synthetic biology is filled with promise, it is essential to navigate the ethical landscape with care. As advancements unfold, we must remain vigilant about the implications of our work. What responsibilities do we hold as scientists and global citizens in ensuring that our creations benefit society as a whole? How do we address the potential for misuse of synthetic biology technologies? These questions invite us to engage in continuous dialogue about the ethical dimensions of our work. Thought leaders in the field, such as Francis Collins, have emphasized the importance of thinking through the implications of our discoveries to ensure that we are not just advancing science, but also upholding the values that guide our humanity. The landscape of synthetic biology is indeed a reflection of our collective aspirations and challenges. The stories of individuals and communities who have embraced collaboration stand as testaments to what can be achieved when we unite behind a common goal. From the researchers who engineer bacteria to produce biodegradable plastics, to those who work tirelessly to develop sustainable agricultural practices, the tapestry of innovation is woven with threads of cooperation and shared purpose. As we envision the future, let us hold on to the belief that our collaborative efforts can lead to solutions that address not only the immediate concerns of our societies, but also the long-term health of our planet. The beauty of synthetic biology lies not just in its scientific achievements, but in its ability to inspire a sense of shared responsibility towards one another and the world we inhabit. In this interconnected narrative, we are reminded that the possibilities are endless when we work together. Each voice, each contribution adds richness to the discourse, propelling us toward a future where synthetic biology serves as a catalyst for social, environmental, and economic progress. What if the next groundbreaking discovery arises from a collaboration that transcends borders and cultures? What if the solutions to our most pressing challenges lie in the hands of those who come together, united by a vision for a better world? The answers to these questions are still unfolding, guided by the collective desire to harness the potential of synthetic biology for the benefit of all. In the spirit of collaboration, the road ahead is illuminated by the shared commitment of scientists, policymakers, and communities working in harmony. By embracing our interconnectedness, we can transform challenges into opportunities, nurturing a global community poised to advance synthetic biology in ways that are ethical, equitable, and sustainable. The journey continues, with each step forward reflecting the enduring belief that together, we can shape a future that resonates with hope, innovation, and shared prosperity. Chapter 10. Personal Engagement. Becoming part of the synthetic biology movement. Imagine standing at the precipice of possibility, where each individual holds the power to influence the trajectory of synthetic biology. The beauty of this field is not solely confined to laboratories filled with advanced equipment and brilliant minds. Instead, it thrives on the contributions of people from every walk of life. Whether you are a student, a hobbyist, or a professional, there are countless ways to engage with this revolutionary movement. The question arises, how can one become part of this transformative journey? One of the most accessible pathways for individuals to contribute is through education. The realm of synthetic biology is expanding rapidly, and educational resources are abundant. Many universities now offer specialized courses in synthetic biology, inviting learners to dive into the core principles that govern this fascinating field. Imagine a classroom buzzing with enthusiasm, where students explore the intricacies of gene editing, synthetic circuits, and the ethical considerations that accompany these technologies. Online platforms have also emerged, providing free or low-cost courses that empower anyone with an internet connection to learn at their own pace. Websites dedicated to open education offer modules that cover everything from the basics of molecular biology to the complexities of biodesign. Furthermore, engaging with local institutions can provide invaluable experiences. Many community colleges and research institutions host workshops and seminars focused on synthetic biology. These gatherings create opportunities for hands-on learning and collaboration. Picture yourself in a lab, working alongside passionate scientists and fellow learners, experimenting with microorganisms and observing their behaviors. Such experiences not only deepen understanding, but also foster a sense of community among those who share a common interest in the potential of biology. As the fabric of synthetic biology expands, citizen science projects have emerged as a vital thread. These initiatives invite individuals to participate in scientific research, often contributing to projects that address local and global challenges. Imagine a community coming together to monitor the health of local ecosystems through the collection of water samples, analyzing them for pollutants and sharing findings with scientists. Citizen science projects like these not only generate valuable data, but also empower individuals to take ownership of their environment. They create a bridge between professional scientists and the public, fostering a collaborative spirit that enriches both parties. Grassroots initiatives are also pivotal in the synthetic biology movement. Many organizations work tirelessly to raise awareness and advocate for sustainable practices. These groups often provide resources and training for those interested in starting their own projects. Imagine a local group of enthusiasts gathering in a community space, exchanging ideas and resources, as they plan to engineer a strain of bacteria capable of breaking down plastic waste. These grassroots efforts exemplify how individual passion can coalesce into powerful movements that challenge conventional practices and inspire change. In addition to direct involvement, inspiring a new generation of synthetic biologists is a crucial component of personal engagement. Encouragement and mentorship can ignite a spark in young minds, leading them to explore the wonders of biology. Schools and educational programs that integrate synthetic biology into their curricula help cultivate curiosity and foster a love for science. Picture a classroom filled with eager students, their eyes lighting up as they design their own experiments, guided by enthusiastic teachers who share their passion for biological innovation. Moreover, outreach programs that connect scientists with local schools can further bridge the gap between academia and the community. Imagine a scientist visiting a classroom, sharing stories of their research adventures and the discoveries made along the way. Such interactions can demystify science, making it more relatable and inspiring students to envision themselves as future contributors to the field. As individuals rally behind the synthetic biology movement, it is essential to recognize the impact of diversity in this space. Every voice adds a unique perspective to the conversation, enriching the collective understanding of how to best utilize synthetic biology for the benefit of all. When people from different backgrounds come together, they share distinct experiences and ideas that can lead to innovative solutions. This commitment to inclusivity is echoed in various organizations that prioritize diversity in STEM fields. They actively seek to empower underrepresented groups, providing scholarships, mentorship programs, and networking opportunities. Imagine a network of aspiring scientists, engineers, and thinkers united in their quest to push the boundaries of what is possible in synthetic biology. The narratives of individuals who have carved their paths in synthetic biology serve as powerful inspirations. Take, for instance, the story of a young woman who, after participating in a local citizen science project, found her calling. She went on to pursue a degree in biotechnology and now works on developing sustainable agricultural solutions. Her journey exemplifies the profound impact that personal engagement can have, not just on an individual, but on entire communities and industries. As we explore the avenues for personal engagement in synthetic biology, it becomes clear that the movement is not solely reserved for scientists or researchers. It is a collective endeavor that invites everyone to participate, regardless of their background or expertise. Each contribution, no matter how small, holds the potential to spark change and inspire others. Imagine a world where individuals unite to address pressing issues such as climate change, food security, and health crises through the lens of synthetic biology. This vision is not merely a dream, it is a reality that is taking shape as more people engage with the field. By fostering curiosity, sharing knowledge, and collaborating across boundaries, we create a vibrant community that thrives on innovation and shared purpose. As we think about the future of synthetic biology, it is essential to cultivate a sense of responsibility and stewardship. With great power comes great responsibility, and as individuals engage more deeply with the field, they must also consider the ethical implications of their actions. How can we ensure that our advancements align with societal values and promote the well-being of all living beings? This question invites reflection and discussion among those involved in synthetic biology. By prioritizing ethical considerations in educational programs and community initiatives, we can help shape a new generation of synthetic biologists who are not only skilled in their craft, but also attuned to the broader implications of their work. The journey of personal engagement in synthetic biology is an ongoing one, filled with opportunities for discovery, collaboration, and inspiration. As more individuals step into this realm, they contribute to a growing movement that challenges the status quo and envisions a future where technology and nature coexist harmoniously. So how will you engage with this vibrant field? Whether through education, citizen science, grassroots initiatives, or mentorship, the possibilities are as vast as the imagination. Each step taken brings us closer to unlocking the full potential of synthetic biology, creating pathways toward a sustainable future that benefits all. The journey into synthetic biology is illuminated by the spirit of collaboration and shared vision. Every individual, no matter their background, can find a place within this dynamic field. Consider the impact of community workshops, where aspiring scientists and seasoned researchers come together to share knowledge and skills. These gatherings create a fertile ground for innovation, enabling participants to experiment and explore ideas that may lead to breakthroughs. Imagine a brainstorming session where diverse perspectives converge, sparking creativity and leading to solutions that none could have achieved alone. Moreover, the rise of online forums and social media platforms has transformed how individuals connect and collaborate. These digital spaces allow for the exchange of ideas and resources across geographic boundaries. Picture a global network of enthusiasts and experts sharing insights on the latest developments in synthetic biology. This connectivity nurtures a collective intelligence that propels the field forward, encouraging individuals to contribute their unique insights and experiences. As we reflect on the importance of educational pathways, it becomes evident that mentorship plays a vital role in shaping the next generation of synthetic biologists. Mentors serve as guiding lights, providing support and encouragement to those embarking on their journeys. Imagine a young student, filled with curiosity, receiving guidance from an experienced researcher who shares not only knowledge but also passion for the field. This relationship can ignite a lifelong dedication to scientific exploration. In addition, outreach programs targeting underrepresented communities are vital in fostering inclusivity within synthetic biology. These initiatives aim to create pathways for individuals who may not have had access to traditional educational resources. Picture a community event where families gather to engage in hands-on science activities, sparking interest in biology among children and their parents alike. Such experiences can inspire a sense of belonging in the scientific community and motivate individuals to pursue careers in synthetic biology. This call for diversity is echoed in the stories of those who have risen to prominence in the field. Take, for example, a researcher who grew up in a small town where opportunities for scientific exploration were limited. Through determination and the encouragement of a supportive mentor, they navigated the challenges and eventually contributed groundbreaking research in sustainable biotechnology. Their story serves as an inspiring reminder that passion and perseverance can lead to remarkable achievements, opening doors for future generations. As the synthetic biology movement gains momentum, the role of citizen scientists becomes increasingly significant. These individuals, often driven by personal passion, engage in research that contributes to scientific knowledge. Imagine a group of local citizens, equipped with simple tools, embarking on a project to study the effects of pollutants in their environment. Their findings not only provide valuable data, but also foster a sense of stewardship for their community and its natural resources. Grassroots initiatives also play a crucial role in shaping public perception and understanding of synthetic biology. Local organizations often work to demystify complex scientific concepts, making them accessible to everyone. Picture a community gathering where experts simplify the principles of genetic engineering, explaining its potential benefits and risks in relatable terms. Such discussions promote informed engagement and encourage individuals to participate in the ongoing dialogue about the future of synthetic biology. The vision of a sustainable future is not solely the responsibility of scientists. It calls for the involvement of citizens at every level. Each individual can contribute uniquely, whether through advocacy, education, or hands-on projects that address local challenges. Imagine a world where communities come together to create solutions for issues like food insecurity or environmental degradation, harnessing the power of synthetic biology as a tool for positive change. As we consider the broader implications of synthetic biology, we must also acknowledge the importance of ethical engagement. Individuals stepping into this field are called to reflect on their responsibilities, not just as scientists, but as members of society. Imagine the conversations that could arise from community forums dedicated to discussing the ethical dimensions of synthetic biology. Such dialogues can cultivate a culture of accountability, where individuals are encouraged to think critically about their contributions and the impacts they may have. Inspiring a new generation of synthetic biologists is not merely about imparting knowledge, it is about fostering a mindset that values curiosity, creativity, and ethical consideration. Educational institutions have the opportunity to cultivate an environment where students feel empowered to ask questions and challenge the status quo. Picture a classroom where students engage in project-based learning, designing experiments that tackle real-world problems. This hands-on approach allows them to connect theory with practice, nurturing a sense of agency and responsibility. Moreover, the role of technology in education cannot be overlooked. Online platforms and virtual laboratories have opened new avenues for learning, making synthetic biology accessible to a broader audience. Imagine students from diverse backgrounds collaborating on projects through digital platforms, sharing insights and discoveries that transcend geographical limitations. This interconnectedness fosters a sense of global citizenship, where aspiring scientists recognize their role within a larger community. As the synthetic biology movement continues to evolve, personal engagement remains a cornerstone of its success. Each individual's journey contributes to a collective narrative of innovation and resilience. Imagine a future where synthetic biology is not just a field of study, but a vibrant community of passionate individuals working together to address humanity's most pressing challenges. The possibilities are limitless, and the contributions of each person matter deeply. So as you reflect on your own potential role in this movement, consider the unique gifts and perspectives you bring. Whether through education, outreach, citizen science, or grassroots initiatives, your engagement can shape the future of synthetic biology in profound ways. How will you connect with this beautiful tapestry of science and community? The answers lie within your imagination, waiting to be realized in the world around you.